Welcome to the Sleepy Scientist. This is the quiet place where science takes its time, where curiosity can unfold at a gentle pace, and where the mind is free to drift. Tonight's exploration turns inward to the smallest stage imaginable, the life of a single cell within the human body. While the body rests, a cell continues its tireless work, listening, repairing, and keeping the rhythm of life steady. Before the story begins, I'd love to know, where are you listening from tonight? And if this kind of gentle science helps you find rest, please consider leaving a like and subscribing. To those already subscribed, thank you for your steady support. It is the quiet heartbeat that keeps these explorations alive. Now, a small smile before we wander deeper. May this not be so interesting that it robs you of sleep, but if it does keep you awake, at least a few secrets of life's smallest worker may settle comfortably into memory. So as the room grows still, one question rises. What does a single cell truly do in the hush between one heartbeat and the next? Life, at every scale, is a tapestry of order woven from chaos. In the grand sweep of biology, it is easy to marvel at the whole human body, at organs, tissues, and thoughts. Yet hidden beneath that larger view lies the true foundation, the individual cell. Every movement, every idea, every breath rests on the quiet efforts of these microscopic units. A single human cell measures only a few micrometers across, far smaller than a grain of sand, and yet within that tiny space unfolds an entire society of molecules. The membrane forms its shoreline, the cytoplasm its busy streets, the nucleus its library, and mitochondria its lanterns of power. Each part has a task, and each task is essential. From these simple, repeated lives, trillions of cells build the body that carries thought, breath, and dream. This journey follows one such cell through its quiet day, not in bursts or spectacle, but in rhythms, energy gathered, messages passed, repairs made, and balances kept. The setting is constant work without complaint, a pattern of microscopic diligence that continues whether noticed or not. What makes this remarkable is not only the complexity, but the calm in which it is conducted. A cell does not rush. It relies on diffusion, gradients, and subtle forces of physics. Molecules drift until they meet. Enzymes act without fanfare. Signals whisper instead of shout. Out of these small motions, stability emerges, a stability that allows the larger body to live and think. To understand a cell is to step into a world where scale defies expectation. Walls are measured in nanometers, yet they are strong enough to guard identity. Messages are carried in molecules so small they escape the naked eye, yet they steer the life and death of the organism. Time is kept not by clocks but by cycles of synthesis and repair, cycles that have ticked reliably for billions of years. This introduction opens the curtain not with noise but with stillness. It sets the frame for what lies ahead. A close look at the cast of characters inside a single cell, the roles they play, and the harmony that keeps them balanced. Through this lens, biology becomes less a textbook list, and more a quiet performance that never pauses, even in the hush of night. And so the journey begins into the soft interior of one small universe, where order is built molecule by molecule, and where the simplest units of life write the story of existence in silence. Between one heartbeat and the next, silence lingers long enough to imagine another world entirely. In that small pause, where the body seems still, countless lives continue unobserved. Among them is the life of one cell, a world unto itself carrying on in measured rhythm. It does not hum like machinery or ring like clockwork. It whispers in ions, in currents, in chemistry unfolding quietly beneath the surface of awareness. A cell is tiny by any standard. Most human cells measure between 10 and 30 micrometers across, far below what even the sharpest unaided eye could see. A speck of dust looks enormous by comparison, yet the scale belies the richness within. Packed into that space is a bustling community of structures, each with tasks that never cease. Even without grandeur, the cell achieves something profound. It sustains life without pause, each moment a small act of balance and order. To step into this space requires leaving behind the human sense of distance. A single micrometer is 1/1,000,000th of a meter. The thickness of a strand of hair is about 70 micrometers, wide enough to line up several cells shoulder to shoulder. Inside that thin space, chemistry becomes geography. The distance from one end of the cell to the other is significant for molecules that must move by diffusion, jostled randomly by the motion of water molecules. Time itself seems different here, not slow or fast in human terms, but steady, measured in the frequency of collisions, the rate of signals, the tempo of reactions. The border that separates the inside from the outside is only a few nanometers thick. This cell membrane, made of a double layer of phospholipids, is the shoreline of this hidden world. Picture a coast where the outer waters touch the air and the inner waters lap the sand. The membrane is not rigid stone but a living beach, always in gentle motion, proteins bobbing within it like piers and boats drifting along a harbour. From the outside, the shore is decorated with sugars that serve as signs and signals, as if flags flew along the coast to announce identity and purpose. Crossing this boundary is not casual. Each ion, each molecule must find a proper passage, like travellers showing the right documents before entering a new land. Sodium and potassium, calcium and chloride slip through gates made of proteins, some opening only when voltage changes, others only when a signalling molecule knocks at the door. Nutrients are ferried in, wastes are pushed out, and messages are received and relayed inward. The shoreline decides with exquisite selectivity who may enter and who must remain outside. Step within, and the cytoplasm opens into a crowded interior. This is not thin water, but a concentrated solution of proteins, salts, sugars, and nucleotides. Enzymes float within, searching for partners in reactions, while organelles provide specialized rooms for specific kinds of work. Vesicles drift like small parcels sealed for delivery. Motor proteins march along filaments, each carrying cargo to the proper address. Their motion so regular it looks almost mechanical, though it is powered by bursts of adenosine triphosphate. The crowding is not chaos, it is carefully balanced. The concentration of ions inside the cell is not the same as outside. Potassium is abundant within, sodium abundant outside. This difference matters. It creates a potential, a soft electrical charge across the shoreline, a stored tension that can be used to send signals, to bring nutrients in, to move waste out. The difference between inside and outside is the very reason life remains organized. Without it, the cell would lose its balance, and the shore would dissolve back into the sea. Toward the center lies a distinct chamber, the nucleus, wrapped in its own double membrane. Here, DNA coils in long, delicate threads, storing instructions in a language of 4 letters repeated billions of times across the human genome. The nucleus is calm but never idle, its pores open and close to allow messenger RNA to leave, carrying transcripts of the genetic library to the protein-making machinery elsewhere. Within this archive, choices are made not by will but by careful regulation. Proteins that encourage certain pages to open, others to stay shut. The nucleus is less a throne room than a library desk, where instructions are copied steadily and accurately, shaping the daily work of the cell. Elsewhere, mitochondria drift, their folded inner membranes bright with the machinery of energy production. They take in oxygen and the products of food molecules, breaking them down further in a dance of electrons and gradients. The result is energy, packaged as adenosine triphosphate, the universal coin of the cell. Without this quiet minting of energy tokens, none of the other players could act. Mitochondria are more than power plants. They are regulators, balancing calcium and even influencing the timing of life and death for the cell. Though small, they carry their own traces of independence, tiny circles of DNA, relics of an ancient partnership formed over 1 billion years ago when free-living bacteria became partners rather than strangers. Scattered throughout the cytoplasm are ribosomes, the factories that knit proteins from the instructions carried by messenger RNA. They read 3-letter codes and match them to amino acids delivered by transfer RNA, building chains that fold into functional proteins. Some ribosomes float freely, producing proteins that stay inside the cell. Others line the surface of the rough endoplasmic reticulum, making proteins destined for membranes or for export. Each protein has a role, whether structural, enzymatic, or regulatory. The ribosomes do not ask why, they simply build, endlessly and efficiently. All of this activity hums at a scale too small to see, and yet it is not random noise. It is order created by chance guided through structure. Molecules bump into one another constantly, but the conditions inside the cell ensure that useful collisions are frequent enough to sustain life. Concentrations are held steady, temperatures are maintained, the pH rests at a comfortable neutrality. Every parameter is tuned with care, not by conscious decision but by the laws of chemistry shaped by evolution's long trial and error. The atmosphere inside a cell is not frantic. Despite the crowding, there is rhythm. The diffusion of molecules has a natural pace. Motor proteins take their steps with steady precision. Ribosomes read their codes at reliable speeds. Mitochondria adjust output smoothly. Even when responding to stress, the cell does not rush blindly. It shifts gears with measured intent. What might look chaotic in a still picture reveals itself as a calm flow when viewed in motion, like a tide pool that seems random until its rhythms are understood. The scale of wonder here is twofold: the minuteness of the parts and the immensity of their combined effort. A single cell is fragile in size, yet in function it is durable, adaptable, and quietly powerful. Life at this level depends not on brute strength but on subtlety. Gradients of ions, folds of membranes, careful exchanges of information. It is a quiet conversation between chemistry and physics, whispered continuously without pause. And it is here, in the hush between heartbeat and breath, that the cell holds its ground. The shoreline of the membrane keeps the world outside from drowning the inside. The cytoplasm provides a lively interior where parts find their places. The nucleus guards memory and offers instruction. The mitochondria shine lanterns of energy. The ribosomes turn messages into tools. Together, they make existence possible, not with drama, but with constant, steady work. A cell does not need applause. Its triumph lies in persistence, in carrying out every moment of life without spectacle. The wonder is not in what is rare, but in what is repeated, over and over, trillions of times across the body, each one a quiet shore where life keeps its balance in perfect calm. Every story has its central figures, and inside a single cell the cast is both small and endlessly busy. 5 main characters keep life running here, each with a simple job that hides a surprising depth of craft. They are the cell membrane, the cytoplasm, the nucleus, the mitochondria, and the ribosomes. Together they do not shout or compete. They move in quiet coordination, like a well-rehearsed ensemble. Begin with the membrane, the elegant boundary that separates the inside from the outside. This barrier is only a few nanometers thick, yet it is the difference between order and dissolution. Built from 2 layers of phospholipids, it resembles a fluid sheet where oily tails hide inside and water-loving heads face outward. It is flexible, more like a calm shoreline than a rigid wall. Proteins drift within it like boats on a bay, opening as channels, closing as gates, or serving as pumps that carry substances against their natural flow. Sugars decorate its outer face, forming a soft glycocalyx that lets neighbouring cells recognise one another. Cholesterol slips among the lipids to keep the surface neither too stiff nor too floppy, adjusting the texture like a skilled gardener tending soil. The membrane's role is clear: hold the inside together permit useful exchange, and politely refuse what would harm. A sodium ion may be turned away while potassium is welcomed, and a hormone may dock at a receptor, starting a ripple of signals without ever stepping inside. The shoreline is never static. Parts are replaced, proteins shift, and vesicles fuse or pinch away, all while the continuity of the border remains intact. Step past this boundary and the cytoplasm opens up. Here lies the crowded interior, not empty water but a rich broth of proteins, enzymes, salts and sugars. This is where much of the work happens, in conditions carefully maintained to keep chemistry humming. The cytoplasm hosts organelles, supports diffusion, and provides the infrastructure for transport. Motor proteins march along filaments of the cytoskeleton, carrying vesicles of cargo, each step powered by a tiny coin of energy called adenosine triphosphate. Without roads or traffic signals, traffic flows smoothly, a testament to the efficiency of probability and design. Enzymes float until they find their substrates, then catalyze reactions at speeds that transform random collisions into dependable outcomes. The cytoplasm balances structure and flexibility. It is viscous enough to slow molecules down, which helps them meet their partners more often, yet fluid enough to allow rapid rearrangements when the cell changes shape or moves. Think of it as a city square at festival time, busy, crowded, and yet somehow always in order. Anchored within this space is the nucleus, the grand library of the cell. Protected by its own double membrane, the nucleus holds DNA, the long sequences that record the instructions for every protein the cell could ever need. The DNA coils around histone proteins, forming chromatin that is carefully arranged so active genes are more accessible, while quieter regions remain tucked away. The nucleus is no silent vault. Its pores are gateways that regulate traffic, allowing messenger RNA to leave carrying freshly copied instructions, while proteins and signals come in to influence which genes are expressed. Within the nucleus, the nucleolus appears as a darker patch. It is the workshop that assembles ribosomal components before sending them out into the cytoplasm. The nucleus is more than storage. It is governance. It decides, through countless regulatory proteins and signals, which sets of instructions are relevant to the cell's present needs. Every choice balances accuracy with flexibility. Repair enzymes scan for errors, while transcription factors gently adjust emphasis, like a librarian pulling forward the most requested books. The nucleus is calm but never passive, a centre of information always in use. Close by, mitochondria drift, power stations wrapped in two membranes. Their inner surfaces fold into cristae, expanding the area available for the machinery that extracts energy from food molecules. Sugars and fats are dismantled piece by piece, their electrons pass down a chain of proteins, pushing protons across a membrane to create a gradient. The return flow of those protons drives ATP synthase, an enzyme shaped like a turbine, which spins out adenosine triphosphate, the universal energy currency. The elegance of this process lies in its reuse of fundamental physics, gradients, charges and flows harnessed to produce life's fuel. Mitochondria carry their own small genomes, relics of their origin as free-living bacteria billions of years ago. They are semi-autonomous but tightly integrated, fusing and dividing as needed, moving to areas of high demand, and playing a hand in processes as varied as calcium balance and programmed cell death. Their job is indispensable. Without their steady minting of energy coins, the rest of the cell would quickly falter. Completing the main cast are ribosomes, small yet vital factories that turn instructions into products. Each ribosome is made of two subunits, themselves built from ribosomal RNA and proteins. Together, they attach to a messenger RNA and read it 3 letters at a time, each codon matched to a transfer RNA carrying a specific amino acid. Like knitters working with perfect memory, ribosomes link amino acids into chains, forming proteins that fold into exact shapes. Some ribosomes float freely, producing proteins destined to stay inside the cell. Others line the rough endoplasmic reticulum, creating proteins that will be sent to membranes or secreted out of the cell entirely. Speed and accuracy define their craft. If an error slips in, quality control mechanisms halt or recycle the faulty product. The ribosome does not question or hesitate. It builds tirelessly, ensuring that the plans copied in the nucleus become tools ready for use. Together, these 5 characters sustain the life of the cell. The membrane keeps the border secure but welcoming. The cytoplasm provides the bustling interior. The nucleus stores and manages the instructions. The mitochondria fuel the activity, and the ribosomes manufacture the machinery needed for every task. They do not act in isolation. A hormone arrives at the membrane. The nucleus responds with a transcript. Ribosomes translate that transcript, mitochondria power the resulting activity, and the cytoplasm carries it all. The dance is seamless. What makes this cast remarkable is how ordinary their actions appear when stated simply, and how astonishing they are when considered in detail. A border only nanometers thick holds an entire world separate from chaos. A watery interior manages countless reactions without losing track. A library of molecules stores the script of life with enough accuracy to persist across generations. Power plants extract energy from gradients invisible to the eye. Tiny factories knit proteins with precision that would shame the finest craftsman. There is also an understated humour in how relentless they are. Ribosomes never stop knitting, though they do not sip coffee. Mitochondria churn out energy tokens endlessly, yet they never send a bill. The membrane constantly adjusts itself, though it has no architect. The nucleus keeps records with a patience no scribe could rival. The cytoplasm manages more errands than the busiest market square, yet never clogs. Each part has a simple job, but taken together, they maintain the living balance that allows thought, breath, and rest. In the quiet moment between heartbeat and breath, this cast carries on without drama. The shoreline holds, the interior bustles, the library copies, the lanterns shine, the workshops knit. The 5 players continue their roles with steady grace, and life persists, seamless and small, within the space of a single cell. Energy is the quiet currency of life. Without it, even the simplest movement inside a cell would fall still. Proteins would remain unfolded, signals would drift unanswered, and the borders that keep the inside and outside apart would collapse. Energy is not a luxury here. It is the gentle stream that keeps every process moving. The name of this currency is adenosine triphosphate, often shortened to ATP. Each molecule of ATP is a tiny coin of power spent by enzymes, pumps and motors to accomplish their tasks. The beauty of it is that ATP is neither rare nor exotic. It is made constantly, used instantly, and recycled tirelessly, like pocket change that never runs out as long as the mint keeps producing. At the centre of this minting process are the mitochondria. Their folded inner membranes house the machinery that turns food into ATP. Yet the process does not begin there. It starts in the cytoplasm, where glucose, a simple sugar, is broken down step by step in a pathway called glycolysis. Glycolysis does not require oxygen, and it is as old as life itself, a metabolic trick that predates mitochondria by billions of years. In 10 small steps, each catalyzed by a different enzyme, one glucose molecule is split into 2 molecules of pyruvate. Along the way, a modest yield of ATP appears, only a few coins, but enough to keep the cell alive in emergencies. If oxygen is available, the journey continues into the mitochondria. Pyruvate enters and is transformed into a molecule called acetyl-CoA, which then joins a larger cycle often called the Krebs cycle, or the citric acid cycle. This is not a frantic spin, but a steady loop of reactions that release electrons from carbon-based fuel. The cycle itself does not produce much ATP directly. Instead, it prepares the stage by handing those electrons to carriers, molecules called NADH and FADH2, that ferry them to the inner membrane. These carriers are the quiet couriers of energy, moving electrons to the chain that truly generates the power. The electron transport chain, embedded in the inner membrane, is where the major minting of ATP occurs. Electrons, full of energy, are passed from one protein complex to the next, each transfer releasing a small portion of usable power. That power is not wasted. It is used to pump protons from the inner space of the mitochondrion to the area between its two membranes. Slowly, carefully, a gradient is created. Protons accumulate outside the inner membrane, creating both a concentration difference and an electrical charge. It is as though water is being lifted behind a dam, potential energy stored in separation. The final step is elegant in its simplicity. The protons, eager to flow back to their original side, can only return through a protein called ATP synthase. This protein acts like a turbine, spinning as protons flow through it. Each spin drives the creation of ATP from its lower energy form, ADP, by attaching an extra phosphate group. In this way, the cell harnesses the natural drive of particles to spread out and uses it to build its energy coins. Oxygen plays the quiet role of final electron acceptor, combining with the used electrons and protons to form water. Without oxygen, the chain halts, the dam no longer fills, and ATP production falls to the modest dribble of glycolysis. The numbers are striking when considered at scale. A single molecule of glucose can yield around 30 ATP molecules when oxygen is available. Multiply that by the countless glucose molecules consumed each second, and the result is an unending river of energy coins. The average human body turns over its own weight in ATP every day, constantly recycling each coin by breaking it down and rebuilding it. At rest, billions of ATP molecules are used every second just to maintain the basics: ion gradients across membranes, protein synthesis, and the continuous maintenance that keeps cells ready for more demanding tasks. What makes this system soothing rather than chaotic is its balance. The cell does not overproduce ATP, nor does it allow its levels to dip too low. Sensors and signaling pathways monitor the ratio of ATP to ADP, adjusting the pace of glycolysis, the Krebs cycle, and the electron transport chain as needed. If the demand rises, perhaps a muscle contracts, or a neuron fires more rapidly, mitochondria respond by increasing their activity. If demand falls, production eases without fuss. Energy supply follows energy demand with the smoothness of breath following thought. There is something comforting in how this process borrows directly from the physics of gradients. The cell does not invent energy from nothing. Instead, it uses what is already true of the universe. Particles spread out, charges balance, molecules seek lower energy states. Life bends these truths to its will by building structures that channel them toward useful outcomes. The proton gradient in a mitochondrion is nothing more than an imbalance, but when directed through ATP synthase, it becomes the steady hum of power that sustains every heartbeat, every thought, every quiet moment of rest. Even when oxygen is scarce, cells are not left entirely powerless. Some cells can switch to fermentation, a process that allows glycolysis to continue by converting pyruvate into lactate, regenerating the NAD+ needed for the cycle. The yield is small, only a few ATP per glucose, but it can mean survival in temporary shortages. Other cells, like those in yeast, may produce ethanol instead. These are fallback strategies, less efficient but practical, evidence of life's adaptability. For most human cells, though, oxygen is the key to steady, generous energy. ATP itself is not stored in large amounts. The cell relies on continuous production, matching supply with use in real time. This makes energy a flowing currency rather than a hoarded treasure. The spending is constant. Pumps along the membrane moving ions, motors walking vesicles, enzymes folding proteins, ribosomes knitting amino acid chains. Each task takes its coin, uses it, and leaves behind ADP, ready to be recharged. The cycle is relentless yet gentle, a rhythm that matches the pace of the living body. There is an understated elegance in the scale of this system. Billions of tiny turbines spin in mitochondria across trillions of cells, each one powered by electrons stripped from food and carried on by invisible couriers. The process is never seen, never heard, but its results are everywhere. Muscles contract, neurons fire, eyes blink, and lungs expand. Energy does not announce itself. It is the silent sponsor of every act of life. In the hush between heartbeat and breath, the cell's power flows with calm certainty. Glucose is split, electrons are carried, protons are pumped, and turbines spin. The shoreline of the membrane is maintained. The nucleus continues its instructions. Ribosomes knit their proteins, and the cell persists. Energy moves without spectacle, shaped by chemistry and guided by structure, delivering just enough, just in time to keep life in balance. The miracle is not the complexity, but the quiet consistency, a process repeated endlessly, perfectly, and without pause, ensuring that the smallest players in the body never run out of strength. Every cell, no matter how small, needs structure. Without it, the interior would collapse into a shapeless soup, and movement would be little more than molecules drifting aimlessly. Instead, within the crowded cytoplasm lies a framework so dynamic and so adaptable that it gives the cell both strength and flexibility. This is the cytoskeleton, a network of filaments and fibers that stretches from the shoreline of the membrane to the quiet depths near the nucleus. The cytoskeleton does not resemble the rigid skeleton of an animal. It is more like a system of highways, scaffolding, and ropes, constantly built, rearranged, and dismantled to meet the moment's needs. Its three main players are actin filaments, microtubules, and intermediate filaments. Together, they provide comfort in form, direction in traffic, and resilience against the stresses of daily life. Actin filaments are the most delicate of the three, thin threads only about 7 nanometers wide. Despite their slim build, they are astonishingly versatile. Actin filaments line the inner surface of the membrane, forming a supportive cortex that maintains the cell's shape. They also assemble into bundles or branching networks, giving rise to structures such as lamellipodia and filopodia, extensions that help cells explore their surroundings or crawl across surfaces. In white blood cells, actin's restless remodeling powers the ability to chase down invaders. In developing tissues, actin helps cells migrate into proper positions, laying the groundwork for organs and structures. Actin filaments are dynamic by design. Their ends can rapidly add or lose actin subunits, allowing them to grow, shrink, or reorganize in seconds. This is why cells can change shape so fluidly, squeezing through narrow gaps or extending exploratory fingers into their environment. In a way, actin is the cell's improvisational dancer, always moving, always ready to shift form. Microtubules, by contrast, are the grand highways of the cell. Each is a hollow tube about 25 nanometers wide, built from repeating units of tubulin. They radiate outward from organizing centers near the nucleus, creating long, sturdy tracks that reach to the periphery. Motor proteins such as kinesin and dynin walk along these tracks carrying cargo. Vesicles loaded with proteins, lipids, or signaling molecules hitch rides like passengers on a dependable train system. Kinesin generally walks outward toward the cell's edge, while dynin moves inward toward the center. This division of labor ensures efficient traffic, preventing jams in the crowded cytoplasm. Microtubules are not static beams. They constantly undergo dynamic instability, a process where they grow by adding tubulin at one end, and then suddenly shrink back when the additions stop. This flexibility allows the network to reorganize quickly, which is especially important during cell division, when microtubules form the mitotic spindle that separates chromosomes. In essence, microtubules combine the sturdiness of an engineered bridge with the adaptability of scaffolding that can be erected or dismantled on demand. The The third player, intermediate filaments, offers something different: resilience. These fibers are about 10 nanometers wide and are built from proteins that vary depending on the type of cell. In epithelial cells, keratin provides strength. In neurons, neurofilaments support long axons. In connective tissue cells, vimentin adds stability. Unlike actin filaments and microtubules, intermediate filaments are not especially dynamic. They do not grow and shrink rapidly. Instead, they form rope-like networks that bear mechanical stress. They anchor organelles in place and connect to structures at the membrane, distributing tension across the cell. This is why cells can withstand being stretched, compressed, or jostled without tearing apart. If actin is the dancer and microtubules the highways, intermediate filaments are the braided ropes that hold everything steady when life pushes and pulls. These three systems do not act in isolation. They interact constantly, linking together to create an integrated framework. Actin connects to adhesion proteins at the membrane, allowing the cell to grip surfaces. Microtubules steer vesicles toward those adhesion sites, bringing supplies to strengthen the grip. Intermediate filaments tie the whole structure into a cohesive mesh, distributing forces evenly. The result is not rigidity, but comfort, an organized interior that is strong enough to resist collapse and flexible enough to change shape when the environment demands. The cytoskeleton also provides direction for internal organization. Without it, mitochondria, vesicles, and other organelles would drift randomly. Instead, microtubules act as tracks to position them properly, while intermediate filaments tether them in place. Even the nucleus is anchored by cytoskeletal connections. This spatial order is vital for efficiency. Signals can travel quickly, cargo reaches its destination, and the cell maintains polarity. The distinction between top and bottom, front and back, that is crucial for coordinated function in tissues. Actin, microtubules, and intermediate filaments are also deeply involved in communication. When the cell experiences mechanical stress, such as pressure from neighbouring cells or changes in the stiffness of the surrounding tissue, the cytoskeleton senses these forces and transmits signals to the interior. This process, known as mechanotransduction, influences gene expression, protein synthesis, and even cell fate decisions. A stem cell resting on a stiff surface may be nudged toward becoming bone, while one on a soft surface may turn into fat. The cytoskeleton is not just scaffolding, it is a sensor, reading the physical world and helping the cell respond appropriately. Even in specialized cells, the cytoskeleton finds unique ways to serve. In neurons, microtubules guide the long extensions of axons, ensuring that signals can travel remarkable distances across the nervous system. In muscle cells, actin filaments and associated proteins form the core of the contractile machinery that generates force. In the cells of the skin, keratin filaments create a tough layer that protects against environmental damage. These variations show how the same basic components can be adapted into roles as diverse as transmitting thoughts, moving limbs, or shielding against the elements. There is a quiet humour in how the cytoskeleton achieves its tasks. Motor proteins hauling vesicles walk with what looks like a comically determined stride, one foot in front of the other, never rushing but never pausing either. Microtubules grow and collapse with such dramatic flair that the term catastrophe is used for their sudden shrinking, though in truth it is just part of their natural cycle. Actin filaments endlessly add and remove subunits, as though they cannot decide on a final arrangement, when in reality their constant remodeling is what gives the cell its remarkable adaptability. Even intermediate filaments, usually the calm ones, will reorganize slowly when the cell changes its identity or prepares to divide, proving that even the steadiest ropes have their moments of movement. The importance of the cytoskeleton is clear when it fails. Certain diseases arise from defects in cytoskeletal proteins. Mutations in keratin can make skin fragile, leading to blisters from the slightest touch. Problems with microtubules can interfere with the ability of nerve cells to transport materials along their axons, contributing to neurodegenerative conditions. Errors in actin regulation can disrupt immune cells, weakening the body's ability to chase down infections. These examples highlight how fundamental the cytoskeleton is to life. Without its comfort and order, the cell cannot perform its daily tasks, and yet, when it functions as intended, the cytoskeleton does so without fanfare. It holds the shape, moves the cargo, senses the environment, and distributes stress, all quietly, or continuously. It does not need to be rigid to be strong, nor frantic to be adaptable. Instead, it embodies a balance that life depends upon, a framework flexible enough to allow change, sturdy enough to provide security, and smart enough to listen to the world outside. Within the hush of the cell, the cytoskeleton offers the comfort of structure. The actin filaments shape the edges and explore the environment. Microtubules stretch like highways to connect distant points, and intermediate filaments weave strength through the whole. Together they provide the architecture of calm persistence, ensuring that the cell is not just a bag of molecules adrift in fluid, but a living, ordered space, capable of withstanding stress while remaining ready to adapt. It is scaffolding that breathes, highways that grow and vanish, ropes that hold fast, all working in harmony to give the cell both stability and grace. Inside the calm chamber of the nucleus lies the storehouse of life's instructions, a vast library written in the language of DNA. Every human cell, with a few exceptions like red blood cells, carries a full set of this library, the entire genome. It contains over 3 billion letters arranged in sequences that form genes, and those genes describe how to make the proteins that carry out nearly every task in the body. The challenge is not in having the instructions, but in knowing when to read them and how to turn them into something useful. The cell cannot afford to rush this process. Accuracy matters more than speed, and elegance comes from precision rather than haste. The journey begins with DNA itself. Each strand is a double helix, 2 complementary chains of nucleotides wound together. One strand carries a sequence that can be read, while the other acts as its mirror image, ensuring that information can be faithfully copied. Genes are not scattered randomly, but are arranged along chromosomes, each with its own landscape of regulatory regions. These regions act like bookmarks or traffic signals, telling the cell where a gene begins, when it should be read, and when it should be left quietly closed. Most of the DNA does not code directly for proteins, but even the non-coding sections have roles in regulating how the important passages are used. When the cell requires a particular protein, the process of transcription begins. Transcription is the act of copying the instructions from DNA into a more mobile format. Messenger RNA. An enzyme called RNA polymerase performs this task. It attaches to the beginning of a gene at a region known as the promoter, prees open the DNA double helix, and begins building a strand of RNA by matching nucleotides to the template strand of DNA. Each adenine pairs with a uracil, each cytosine with a guanine, and each guanine with a cytosine, producing a complementary copy of the gene sequence. This RNA copy is not a permanent book taken from the library, but more like a photocopy of a single page. The DNA remains safe in the nucleus, while the RNA carries the instructions outward to the cytoplasm where proteins are built. In this way, the master library is never exposed to unnecessary wear or damage. Only the copies travel, and they are designed to be temporary, used for a time and then degraded when no longer needed. The making of RNA is a carefully guided affair. Proteins called transcription factors help determine which genes should be transcribed at any moment. Some transcription factors respond to signals from outside the cell, such as hormones or growth factors. Others respond to conditions inside, like the availability of nutrients or the presence of stress. The combined effect is a subtle governance system, one that ensures the right genes are turned on at the right time. The process is not frantic. A cell may only use a fraction of its genes at any given time, tuning its activity to match its role and environment. A skin cell expresses different sets of genes than a neuron, not because they carry different DNA, but because they choose different pages to copy. Once the initial RNA copy is made, it undergoes refinement. In eukaryotic cells, the first draft is called a primary transcript, and it contains more than the cell ultimately needs. Sections called introns are removed in a process known as splicing, while the useful parts, called exons, are joined together to form the final message. At the same time, the RNA receives a protective cap at its beginning and a long tail of adenine nucleotides at its end. These modifications help stabilize the molecule, guide it out of the nucleus, and regulate how long it survives in the cytoplasm. Splicing also allows for flexibility. A single gene can be spliced in different ways to produce slightly different proteins, giving the cell more variety from the same basic script. Once the messenger RNA is ready, it leaves the nucleus through the nuclear pores. These pores are large protein complexes that act like selective gates, allowing RNA to pass out while maintaining the integrity of the nuclear environment. The RNA then drifts into the cytoplasm, where ribosomes will read its sequence and assemble the corresponding protein. This marks the transition from information storage to action, from quiet pages to working tools. The calmness of this process belies its importance. Errors in transcription can have significant consequences. A single incorrect nucleotide may alter the instructions enough to produce a faulty protein. To reduce such risks, proofreading mechanisms and quality control checkpoints are in place. RNA polymerase itself can backtrack and correct mistakes, while specialized surveillance systems detect faulty RNA and degrade it before it causes harm. These safeguards ensure that the information flowing out of the nucleus remains reliable, keeping the cell's operation steady. The scale of transcription is impressive. A single active gene may produce thousands of RNA copies, each leading to the production of many proteins. At the same time, other genes may sit quietly for years, waiting for the right signal to awaken. Some of these genes may never be used in a given cell type, locked away permanently as part of the cell's specialization. Others are called into action only during stress, repair, or developmental changes. The genome is not a static set of instructions, but a responsive archive, ready to adjust to the needs of the moment. There is an understated grace in how information flows without rush. DNA remains safely tucked away, its double helix coiled and compacted to protect it from damage. RNA polymerase moves along a gene at a steady pace, not hurried, not lazy, but methodical. Splicing reshapes transcripts with patient accuracy. Nuclear pores release the finished messages as if they were letters slipping quietly through a postbox. Each step is designed not for speed, but for fidelity. Even humour can be found in the patience of the system. Imagine a massive library where most of the books remain on the shelves, but certain passages are copied again and again depending on the needs of the day. The librarians are tireless, the copying machines precise, and the shelves always kept in perfect order. This cell does not read its entire genome at once. That would be like trying to read every book in a library simultaneously. Instead, it samples thoughtfully, producing only the excerpts required, and doing so with such calm that the whole process feels almost serene. The connection between DNA and RNA is the foundation of the central dogma of molecular biology, the guiding principle that information flows from DNA to RNA to protein. This directionality preserves stability. The library is never worn out because it is not carried into the noisy marketplace of the cytoplasm. Instead, the information is transcribed into flexible copies that can move, interact, and eventually be discarded without harm. In this way, the cell maintains both permanence and adaptability. Permanence in its stored DNA, adaptability in the countless RNA transcripts it produces on demand. In the stillness of the nucleus, life's instructions are managed without fanfare. DNA holds the blueprint, transcription makes temporary copies, and RNA travels outward to guide the making of proteins. The process is so familiar to the cell that it unfolds with a quiet rhythm, day and night, heartbeat after heartbeat. The marvel lies not in haste, but in the steady assurance that the right messages will be delivered when they are needed. From storehouse to action, the flow of information is calm, precise, and utterly reliable, a system designed for continuity rather than speed. It is biology's way of saying that order does not require hurry. Sometimes the best results come from a pace that values accuracy above all else. Inside the cell, construction is constant. Proteins, the workhorses of life, must be built with exquisite care. They are enzymes that guide reactions, structural elements that give tissues shape, channels that allow ions to pass, and messengers that carry signals from one place to another. Each one is unique, yet all are made from the same raw materials, chains of amino acids. The elegance lies not just in making these chains, but in folding them into the precise three-dimensional shapes that allow them to function. And because even a small mistake can lead to disaster, the cell has evolved a system of quality control that would put the most careful assembly line to shame. The story begins with ribosomes, those tireless factories scattered across the cytoplasm and studding the surface of the rough endoplasmic reticulum. Each ribosome attaches to a strand of messenger RNA and reads its code 3 letters at a time. Each triplet specifies one of 20 amino acids, which are delivered by transfer RNA molecules. With mechanical precision, the ribosome links the amino acids together in sequence, forming a growing chain. It is a process that looks simple, but is astonishing in scale. A ribosome can add several amino acids every second, never skipping, never losing its place. The result is a linear chain, like beads strung on a necklace, but unlike jewellery, this necklace must twist and fold into an exact shape, or it cannot work. This folding is where the cell's real craftsmanship begins. Some proteins fold spontaneously as soon as they leave the ribosome, guided by the chemistry of their own amino acids. Others need help from specialised chaperone proteins, which prevent incorrect interactions and provide a sheltered space for folding. Chaperones are not permanent parts of the protein. They act like mentors, guiding the process and then stepping aside when the job is done. If folding goes awry, the protein may clump or misfunction, which can be harmful. To prevent this, misfolded proteins are often tagged for recycling, broken down into their component amino acids so the raw materials can be used again. Efficiency and accuracy remain the guiding principles. Many proteins are destined for particular locations such as the cell surface, the lysosomes, or for export out of the cell entirely. These proteins begin their journey on ribosomes bound to the rough endoplasmic reticulum. As the ribosome builds the chain, the emerging protein is threaded directly into the lumen of the ER, where folding begins with the assistance of resident chaperones. Here, additional modifications take place. Sugars are added in a process called glycosylation, which not only helps proteins fold correctly, but also serves as an address label for later delivery. Disulfide bonds may be formed between certain amino acids, adding stability to the final shape. In the ER, folding and editing are relentless. Proteins that meet the standard continue onward. Those that fail are held back and eventually dismantled. This is quality control at the molecular scale, ensuring that only well-crafted products leave the factory. Once a protein has passed its first inspection, it is packed into a vesicle and sent to the Golgi apparatus, a series of flattened membrane sacs often compared to a postal sorting centre. In the Golgi, proteins are refined further. Sugars attached in the ER may be trimmed, altered, or expanded into more complex structures. Additional tags are added, guiding proteins to their ultimate destinations. Some are marked for secretion, others for the plasma membrane, and still others for organelles like lysosomes. The Golgi works with quiet efficiency, receiving vesicles from one side, processing their contents step by step, and then shipping them out from the other. It is not hurried. The rhythm is steady, with vesicles budding, fusing and departing in a continual flow. From the Golgi, vesicles carry their cargo along cytoskeletal tracks to the proper addresses. Motor proteins haul the vesicles with steady strides, using ATP as fuel. A vesicle destined for the cell surface will eventually reach the membrane and fuse with it, releasing its protein cargo into the extracellular space or embedding it into the membrane itself. Others may be directed inward, fusing with lysosomes where their proteins perform digestive work. The targeting is remarkably accurate, guided by molecular signals that act like shipping labels, ensuring that each delivery reaches the correct location. The importance of this careful process becomes clear when it fails. Misfolded proteins that escape quality control can accumulate into harmful aggregates, linked to conditions such as Alzheimer's disease or cystic fibrosis. Errors in trafficking can misplace enzymes, leaving lysosomes unable to digest properly or cause secreted proteins to be trapped inside the cell. These examples highlight just how crucial accuracy is. The cell's energy is well spent on proofreading, editing, and regulating, because the consequences of error ripple outward far beyond the cell itself. There is a kind of a light humour in how meticulous the process is. The ribosome, lacking any awareness, churns out chains of amino acids with perfect reliability. Chaperones fuss over folding like careful tailors, making sure each seam lies flat. The ER catches mistakes with the vigilance of a quality inspector who never sleeps. The Golgi decorates and redirects with the quiet competence of a shipping clerk who always knows which parcel goes where. None of these parts think, yet together they achieve a level of accuracy that human engineers can only admire. The scale at which this unfolds is staggering. A single cell may produce millions of proteins every hour. Each one is built from a sequence of amino acids that must be assembled in order, folded properly, modified as needed, and delivered to the correct site. Most proteins have lifespans of only hours or days before they are broken down and replaced. This constant turnover means the assembly lines never rest. The cell's interior is a factory that runs continuously, not with clanging machines but with silent molecular choreography. The beauty of protein making lies not only in its complexity, but also in its calm consistency. It is a process that could be frantic, but instead it is steady and orderly. Each step follows naturally from the one before, and every checkpoint ensures that the result is usable. From the first amino acid joined at the ribosome to the final vesicle fusing with its destination, the journey is seamless. The proteins emerge ready for work, whether that work is catalyzing reactions, forming scaffolds, carrying signals, or defending the cell from stress. In the stillness of the cell, construction never stops. Chains of amino acids form, fold, and refold. Chaperones watch with patient care, and vesicles glide along invisible roads to deliver their cargo. The ER inspects, the Golgi refines, and the membrane receives. Proteins appear not in bursts but in a continuous stream, each one a small act of precision. It is this careful making, repeated countless times, that sustains life quietly and reliably, ensuring that the machinery of the cell is always ready, always balanced, and always crafted with patient attention. Every cell is both a listener and a communicator. It exists within a body where trillions of neighbours are carrying on their own tasks, and without coordination chaos would quickly take over. Signals flow in and out constantly, guiding growth, movement, repair, and countless other activities. These signals do not shout or blare. They whisper, arriving as subtle changes in molecules that drift across the fluid environment. Timing matters just as much as the message itself, because a signal that comes too late or lingers too long can be as disruptive as one that never arrives at all. At the surface of the cell, receptors stand like watchful gatekeepers. They are proteins embedded in the membrane, their shapes tuned to recognize specific molecules. Some bind to hormones carried in the bloodstream, others respond to neurotransmitters released from nearby cells, and still others detect nutrients, light, or even mechanical pressure. The principle is always the same. A signal molecule fits into the receptor like a key into a lock, and when it does, the receptor changes shape. That shift, though tiny, is enough to start a cascade inside the cell. One of the gentlest examples can be found in the way cells respond to adrenaline. When this hormone binds to its receptor on the cell surface, it triggers a chain of events inside. The receptor nudges a protein called a G protein, which then activates an enzyme that produces a molecule called cyclic AMP. This molecule, though small and short-lived, spreads the word quickly by activating a set of enzymes that change the behavior of the cell. The result might be the breakdown of glycogen into glucose for energy, or an increase in heart muscle contraction. From the outside, all that is visible is a faster heartbeat or heightened alertness, but deep inside, it is a graceful dance of molecules responding to a signal. Signals are not always about excitement or emergency. Many are about balance. Insulin, for instance, carries the calm message that glucose should be stored rather than released. When insulin binds to its receptor, it initiates a pathway that moves glucose transporters to the surface of cells, allowing sugar to flow inward. It is a gentle invitation, not a command shouted through the system, and the result is stability in blood sugar levels. When this process falters, as in diabetes, the consequences ripple through every cell, showing how crucial these conversations are. Inside the cell, signals are often carried by what are called second messengers. These molecules amplify the initial message from the receptor, ensuring it is not lost in the noise of the cell's busy interior. Calcium ions, for example, can be released from internal stores in response to a signal. Even a small pulse of calcium can activate proteins that drive muscle contraction, trigger vesicles to release neurotransmitters, or spark gene expression. Because calcium is so powerful, its levels are tightly controlled, with pumps and buffers ensuring that each pulse is brief and precise. It is not the amount of calcium alone that matters, but the timing and rhythm of its release. This precision is one of the most fascinating aspects of cellular signaling. A single molecule like calcium or cyclic AMP can mean different things depending on how long it stays around, how much is present, and in which part of the cell it appears. A brief spike in calcium might trigger a heartbeat, while a sustained wave could lead to changes in gene activity. The cell interprets these subtle differences as carefully as a musician reads notes on a page. It is not just the note itself, but its duration and placement in the melody that create meaning. The complexity deepens when signals overlap. A cell rarely receives just one instruction at a time. Dozens of receptors may be activated simultaneously, with their cascades weaving together inside. Some reinforce each other, while others compete. This integration allows the cell to make decisions, balancing conflicting inputs to choose the most appropriate response. It is much like standing at a busy intersection, hearing multiple voices at once, and still managing to understand what is most important to act upon. There is also a remarkable feedback built into these systems. After responding to a signal, many receptors become less sensitive for a while, a process called desensitization. This prevents overstimulation and ensures that the cell does not overreact to prolonged signals. In other cases, the cell may increase the number of receptors, making itself more responsive if signals are rare. This adaptability shows how dynamic the system is, constantly adjusting its sensitivity to maintain balance. Communication does not only flow inward. Cells also send signals outward, releasing molecules into their surroundings to influence others. Nerve cells release neurotransmitters in precise bursts. Muscle cells secrete growth factors when they need repair, and immune cells release cytokines to call reinforcements. Each signal has its own context, its own meaning, shaped not just by the molecule itself, but by where and when it is delivered. In multicellular life, this exchange of messages creates harmony. A wound on the skin is healed because cells at the site release signals that attract immune cells, stimulate blood vessel growth, and encourage nearby cells to divide. Muscles grow stronger in response to exercise because stressed fibers release signals that promote repair and adaptation. Even the rhythms of sleep and wakefulness are governed by hormones like melatonin, circulating in predictable patterns that guide cells across the body. The beauty of all this lies in how quiet it is. No cell ever raises its voice. Molecules drift, bump gently into receptors, and dissolve back into the fluid when their work is done. The cell listens, interprets, and responds with measured steps. Timing is everything, from the millisecond pulses of calcium in a neuron to the steady daily rise and fall of melatonin. Too fast, and the message is missed. Too slow, and the opportunity is lost. The balance must be just right. Life itself depends on these signals. Without them, a cell would be deaf to its environment, blind to its neighbours, and unable to coordinate with the larger whole. With them, trillions of cells cooperate, each tuned into the constant flow of information, adjusting their behaviour in ways that make the body function smoothly. It is not a frantic process, but a serene one, a conversation carried out in whispers, with timing as precise as a conductor's baton. When viewed in this way, cellular signalling becomes less like a machine and more like a symphony. Each receptor is an instrument, each second messenger a note, and each pathway a melody. The whole performance is layered, delicate, and endlessly variable, yet always coherent. It is not rushed, nor is it chaotic. It is a gentle unfolding of messages that allow a cell to stay in tune with its world, and through that tuning, the larger organism remains alive, balanced, and quietly thriving. The boundary of a cell is not a wall, nor is it a flimsy soap bubble. It is something in between, a flexible shoreline where the inner world of the cell meets the outer sea of the body. This delicate border is the cell membrane, a layer so thin it cannot be seen without powerful magnification, yet it holds the power to decide what comes in, what goes out, and what must remain steady. To think of it is to imagine the hush between heartbeat and breath. A pause where tides shift gently, carrying molecules like grains of sand. The membrane itself is made of a double layer of lipids, tiny molecules that resemble matchsticks with a head and a tail. The heads are drawn to water, while the tails shy away from it. When arranged together, they form a neat bilayer, with the water-loving heads facing outward to the cell's watery environment and inward toward the cytoplasm, while the water-avoiding tails press together in the middle. This arrangement creates a barrier that is both stable and dynamic, a surface that is fluid and alive. It does not keep everything out, but instead manages the flow with quiet precision. Some molecules slip through this boundary as if by chance. Oxygen and carbon dioxide, both small and uncharged, drift across with ease, following the natural gradient from areas of higher concentration to lower concentration. This simple movement, known as diffusion, requires no energy. It is the molecular equivalent of rolling downhill, matter taking the easiest path, spreading out until balance is reached. It happens invisibly, constantly, and it ensures that every cell is supplied with oxygen and relieved of carbon dioxide without any conscious effort. Water, too, moves with this passive grace, guided by a process called osmosis. If one side of the membrane has more dissolved particles, water will flow toward it, seeking to even out the concentration. It is as if the cell were adjusting its internal tides, allowing water to flow in or out depending on the needs of the moment. This movement is not dramatic but steady, a quiet, balancing act that keeps the cell from swelling too large or collapsing in on itself. Not every substance can wander freely. Many require passage through channels or gates embedded in the membrane. These channels are proteins, each shaped to allow only certain molecules to pass. Some open only when triggered by a signal, while others form continuous tunnels. Imagine them as doorways in a long seawall, each one admitting a particular kind of traveller. A sodium ion may need one door, while potassium requires another, and glucose yet another. This selectivity keeps the cell's chemistry finely tuned. Ion channels are among the most fascinating of these gates. They control the movement of charged particles like sodium, potassium, calcium, and chloride. Though tiny, these ions carry electric potential, and their controlled movement generates signals that power nerves and muscles. In neurons, waves of ions flowing through these channels create the pulses that allow thoughts, memories, and reflexes to exist. The timing of each channel's opening and closing is measured in milliseconds, yet the effects ripple outward into everything from a heartbeat to the blink of an eye. Transport across the membrane is not always passive. At times, the cell must move substances against their natural gradient, pushing them uphill instead of letting them drift downhill. This requires energy, usually in the form of ATP. Pumps within the membrane take on this task, binding to ions or molecules, burning a small amount of energy and shuttling their cargo across. One of the most crucial examples is the sodium-potassium pump, which tirelessly maintains the balance of these ions across the membrane. This balance is essential for electrical signaling, nutrient absorption, and countless other processes. The membrane is not only a filter but also a communicator. Receptors embedded within it catch messages from hormones, neurotransmitters, and other signals, translating the outside world into internal changes. Channels themselves can also act as sensors, opening only in response to changes in voltage, pressure, or the binding of a particular molecule. In this way, the boundary of the cell is not silent, it is constantly listening, responding, and adapting. There is also elegance in how the membrane reshapes itself to move larger items. Endocytosis is the term for when the membrane folds inward, engulfing material and pulling it into the cell. This might be nutrients, signaling molecules, or even entire microorganisms. The reverse, exocytosis, occurs when the cell packages something for delivery, pushing it outward through the membrane. Nerve cells, for example, rely on exocytosis to release neurotransmitters into the gaps between them, ensuring communication across the nervous system. These processes are not clumsy but highly orchestrated, more like a tide retreating and advancing than a clumsy shove. The idea of gradients, differences in concentration, pressure, or charge, is at the heart of these exchanges. A gradient is like a slope down which molecules roll, or a pressure that pulls them forward. Life depends on maintaining these gradients, keeping inside and outside distinct yet connected. Without them, the cell would lose its spark, collapsing into equilibrium where no work could be done. With them, the cell remains alive, charged, and ready to respond. On a larger scale, the cell membrane mirrors the balance of all living things. It is neither an impenetrable fortress nor an open floodgate. It allows flow without losing form, connection without surrendering identity. Each exchange is deliberate, whether a whisper of oxygen passing through, a pulse of ions firing a neuron, or a vesicle releasing its contents into the world. The tides of exchange at this cellular shoreline are ceaseless but calm. They do not roar or crash, but instead lap steadily, maintaining the delicate balance of life. Boundaries are essential not because they divide, but because they create the possibility of interaction. In every moment, across every cell, molecules drift, flow, pump, and pulse, keeping the rhythm of life in motion, quiet as the hush between a heartbeat and a breath. A single cell taken on its own is a marvel of structure and activity, but very few cells ever exist in true solitude. Life thrives not just in individuality but in community, and the body is a living testament to that truth. Cells are rarely loners, they are social beings, constantly interacting with neighbours, with their surroundings, and with the vast cooperative networks that make up tissues and organs. The health of one depends on the collective, and the collective depends on each individual cell holding up its role. To begin, consider the environment that surrounds most animal cells. They are not left to float aimlessly in an empty void. Instead, They rest in and attach to a supportive meshwork called the extracellular matrix. This is not simply background filler. It is a rich and dynamic network of proteins and carbohydrates that gives tissues their structure, much like scaffolding supports a building. Collagen, one of the most abundant proteins in the body, forms sturdy fibers that lend strength and resilience. Elastin, as the name suggests, adds stretch and flexibility, allowing tissues to bend and move without tearing. Glycoproteins, decorated with branching sugar chains, act as bridges between cells in this supportive framework. Together, the extracellular matrix forms a kind of connective landscape, keeping cells in place while also guiding their behavior. This external matrix is not rigid. Cells actively interact with it, sensing its composition, remodeling it when necessary, and even using it as a kind of language. For example, when skin is wounded, cells can detect changes in the matrix around the injury. Fibroblasts rush in to produce new collagen, while immune cells respond to chemical signals embedded in the damaged environment. The extracellular matrix, then, is not just a supportive cushion, but a communicator, providing context and cues that shape how cells behave. Cells also establish direct relationships with one another through specialized junctions. These connections allow them to exchange materials, share signals, and create barriers. Think of them as different kinds of bridges, each with its own design and purpose. One of the most straightforward forms of connection is the tight junction. These act like zippers between neighboring cells, stitching their membranes together so tightly that nothing can slip between. Tight junctions are vital in tissues like the lining of the gut, where the contents of the intestine must stay separate from the bloodstream. Without them, barriers would leak, and harmful substances could sneak through unchecked. Then there are gap junctions, which are quite different. These are tiny tunnels directly connecting the interiors of adjacent cells. Through them, small molecules and ions can pass freely, allowing cells to coordinate their activities with remarkable speed. In the heart, gap junctions are essential. They let ions flow from one muscle cell to the next, ensuring that the heartbeat spreads as a synchronized wave rather than a chaotic spasm. This is communication at its most immediate and vital, carried out in silence at the cellular level. Adherens junctions and desmosomes serve as the anchors of tissues. They connect cells together not just with a casual handshake, but with a firm grip linking their internal scaffolding to one another. This makes tissues resilient, able to withstand stress and stretching. The skin, for example, is held together by countless desmosomes that keep its layers intact despite the daily friction of movement. Without them, tissues would fall apart at the slightest touch. This cooperative architecture highlights a crucial point. Cells are not only part of their community, they depend on it for survival. Many cells require constant signals from their neighbors and from the extracellular matrix to stay alive. When cells lose these supportive interactions, they may undergo programmed death, a process known as apoptosis. In this way, the community acts as both a safety net and a quality check. Cells that drift away or fail to integrate can be gently removed for the greater good. Beyond physical connections, cells also cooperate chemically. They secrete growth factors, hormones, and other signaling molecules into their shared environment, influencing one another's behavior. These signals can encourage growth, trigger division, or call for repair. In the immune system, cells constantly send and receive chemical messages, coordinating complex responses to infection. The harmony of tissues depends on this constant chatter, a conversation conducted in molecules rather than words. One striking example of cellular cooperation is seen in the liver. Hepatocytes, the main liver cells, line up in plates, connected by tight junctions that control what enters the bloodstream. Between these rows, specialized channels carry bile, produced and secreted collectively by the cells. Every hepatocyte contributes, and together they form a functioning organ capable of detoxifying blood, producing proteins, and storing energy. No single cell could achieve this alone. The story is similar in bone. Osteoblasts build new bone, osteoclasts break it down, and osteocytes, the mature bone cells, communicate through long extensions, sensing mechanical stress and directing the balance of construction and destruction. Bone is not simply a hard, lifeless material. It is a living community of cells, maintaining strength and flexibility through cooperation. Even in the brain, where individuality might seem to reign supreme, cooperation is essential. Neurons communicate through synapses, tiny junctions where electrical impulses are translated into chemical signals, but neurons do not work in isolation. They are supported by glial cells, which provide nutrients, maintain the extracellular environment, and even help fine-tune connections. Together, this community of diverse cells allows for thought, memory, and sensation. The theme that emerges is not one of competition, but of cooperation. Cells find their place by linking with others, by building connections both physical and chemical. They shape and are shaped by their community, creating tissues that can move, think, heal, and grow. The extracellular matrix provides the framework, while junctions and signals knit individuals into a whole. One could say that a cell's greatest achievement is not survival in isolation, but survival in harmony. Just as a single musician cannot form an orchestra, a lone cell cannot form a living body. The music of life comes from their cooperation, from the exchange of signals and support across countless boundaries. This quiet, constant collaboration sustains the structure and function of every organ and ultimately of life itself. The cell, then, is not only an individual but a neighbour. Its health depends on its community, and in turn, the community depends on each cell's faithful contribution. Every heartbeat, every breath, every thought is built on this principle of cooperation. Millions of cells bound together not by force, but by connection. Life does not thrive in extremes. It flourishes in balance, in the narrow space between too much and too little. Within the quiet walls of a cell, balance is carefully maintained, even though the world outside can shift unpredictably. This state of balance is called homeostasis. It is not the same as stillness, because stillness would mean lifelessness. Instead, it is a constant, gentle adjustment, like a musician tuning strings while the song continues to play. The music of the cell depends on these adjustments, subtle corrections that keep every process aligned with life. Imagine temperature as one example. Cells, like the organisms they form, work best within a limited range. If conditions rise too high, proteins begin to lose their shape, and with that shape goes their function. If conditions fall too low, movement slows, reactions drag, and energy falters. The cell, however, does not passively accept these shifts. Instead, it activates responses that bring conditions back toward a set point. These set points are like quiet targets, preferred ranges where systems work smoothly. To maintain these targets, cells rely on feedback loops. Negative feedback, in particular, is the soothing mechanism that allows balance to persist. In negative feedback, a change in one direction triggers responses that counteract it, nudging the system back toward equilibrium. Think of it as a lullaby, keeping activity steady without allowing it to spiral out of control. Calcium regulation provides a useful example. Calcium ions are essential messengers, used in muscle contraction, nerve signaling, and countless cellular processes. But the levels of calcium inside the cell must be carefully controlled. If too many calcium ions flood in, signaling pathways may misfire or even trigger cell death. If too few are present, necessary signals cannot be sent. The cell manages this with pumps, channels, and binding proteins that release calcium when needed, and remove it when the level drifts too high. Each adjustment is small, but together they create stability. The same principle applies to glucose, the main energy source for many cells. When glucose levels in the blood rise, cells take in more and store some as glycogen. When levels fall, glycogen can be broken down to release glucose once again. Hormones like insulin and glucagon coordinate this balance, ensuring that cells have enough fuel without being overwhelmed. The cycle is not rigid, it ebbs and flows, but the end result is a range where the cell can continue to function comfortably. The beauty of these feedback systems is that they prevent runaway reactions. Without negative feedback, processes could accelerate uncontrollably. Consider body temperature. If heat triggered responses that created even more heat, the cycle would spiral upward until it caused damage. Negative feedback breaks such spirals by pushing back against extremes. It does not silence change but softens it, guiding the system back to its preferred rhythm. Even within the cell cycle, the process of growth and division, feedback mechanisms guard against imbalance. Proteins check whether DNA has been copied correctly before allowing the cell to move forward. If errors are detected, signals pause the cycle to allow repairs. This checkpoint system protects the integrity of genetic information, ensuring that mistakes are not passed along. The feedback here acts like a gatekeeper, holding the cell in place until conditions are safe to proceed. Homeostasis is not limited to internal chemistry. The cell membrane, acting as a shoreline, also contributes to balance by regulating exchanges with the environment. Ion channels open and close in response to changes, restoring electrical balance across the membrane. Transporters adjust the inflow of nutrients and the removal of waste, preventing either scarcity or buildup. This boundary work is constant, never ceasing, and yet it is so smooth that the cell continues without conscious pause. The marvel of homeostasis lies in its adaptability. The Z points are not fixed in stone but can shift depending on circumstances. For example, during exercise, muscle cells demand more energy. Feedback mechanisms respond by increasing glucose uptake and oxygen delivery, temporarily raising the set point for energy use. When the activity ends, the system drifts back to its resting state. Balance here is not rigid immobility, but a flexible stability, one that allows the cell and the body to adjust to changing needs. Some processes use positive feedback, though these are rarer and more carefully controlled. Positive feedback amplifies change rather than counteracting it, like a snowball rolling down a hill. This can be useful in special cases, such as the rapid release of calcium in muscle cells during contraction. The signal starts small, but is quickly reinforced until the desired action is complete. Positive feedback is powerful, but without limits it could be destructive. That is why negative feedback is the foundation of homeostasis, the calming voice that restores balance once the surge has passed. Cells also use feedback to maintain their volume. Water moves freely across membranes by osmosis, responding to differences in solute concentration. If too much water enters, the cell risks swelling. If too much leaves, it may shrink. To manage this, cells adjust the concentration of solutes inside, pumping ions in or out to counter the movement of water. This invisible dance of molecules prevents the cell from drifting too far from its ideal size, allowing it to remain stable even as its environment changes. Perhaps the most remarkable feature of homeostasis is its constancy over time. The human body maintains its core temperature within a degree or two despite weather, activity, or rest. Cells keep their internal pH within a narrow range, allowing enzymes to work efficiently. Potassium, sodium, and chloride levels are held steady to preserve electrical gradients. Each of these balances depends on continuous feedback loops that never rest. The constancy feels effortless only because the mechanisms behind it are tireless. This idea of balance can be extended beyond the cell itself. Multicellular organisms depend on homeostasis at every level. Organs coordinate with one another, and entire systems work in harmony. But the principles remain the same: detect change, respond, and return toward equilibrium. It is a lullaby repeated across scales, from molecules to tissues to the whole body. To picture it, imagine walking a tightrope. Every step requires adjustment, leaning slightly left, then right, shifting balance forward and back. The goal is not to freeze in place, but to keep moving steadily across. Cells do the same, making countless tiny corrections that keep them balanced while life continues to unfold. Stability is achieved not by resisting movement, but by embracing it with control. In the end, homeostasis is what allows life to persist in a world of constant change. It is the quiet song of balance, playing beneath every heartbeat, every breath, every cellular reaction. It keeps order without demanding rigidity, ensuring that even in the face of fluctuation, life remains steady. The cell's lullaby of feedback ensures that the music continues, soft, steady, and enduring. Cells are not perfect machines. They live in a world full of hazards, shifting temperatures, reactive chemicals, stray radiation, and even the occasional mistake within their own processes. Left unchecked, these hazards could spell disaster, yet life has persisted for billions of years. The secret is that cells are not only builders, but also diligent repair crews, quietly fixing damage as it arises. Their work is constant, often invisible, and it is the reason organisms can endure for decades rather than mere days. Consider DNA, the instruction book of life. Every moment it is under threat. Ultraviolet light from the sun can cause bases to stick together incorrectly. Reactive oxygen molecules generated as byproducts of metabolism can nick or distort the double helix. Even the very act of copying DNA during cell division is risky, with potential for mistakes every time a nucleotide is added. Yet the cell is equipped with sophisticated systems to detect and repair this damage before it spreads. One such system is base excision repair. Imagine a proofreader working through a text line by line. When a single letter is wrong, the proofreader carefully removes it and replaces it with the correct one. Enzymes in base excision repair do just this, scanning the DNA for small, subtle errors. When they find one, they cut out the damaged base, replace it, and seal the backbone as though nothing ever happened. When the damage is larger, another method called nucleotide excision repair steps in. This is more like replacing an entire sentence rather than a single letter. A short stretch of the DNA strand containing the damage is removed, and fresh nucleotides are filled in to match the untouched strand. This process is particularly important for fixing the harmful lesions caused by ultraviolet light, which can distort the helix into unnatural shapes. Sometimes the damage occurs during the copying process itself. DNA polymerase, the enzyme responsible for replication, is remarkably accurate but not flawless. Fortunately, it comes with a built-in proofreading function. When it places the wrong nucleotide, it senses the mismatch, backs up, removes the mistake, and inserts the correct one. If an error slips through, additional mismatch repair systems catch it after replication is complete. These corrections prevent mutations from becoming permanent, preserving the integrity of the genetic code. Of course, not all DNA damage can be fixed. When breaks occur in both strands of the double helix, the cell faces one of its greatest challenges. Two main repair pathways come into play: homologous recombination and non-homologous end joining. Homologous recombination is precise, using an undamaged copy of the DNA as a template to restore the sequence. Non-homologous end joining is faster but riskier, stitching the broken ends together directly. While it may leave small errors, it prevents the break from destabilizing the chromosome entirely. These processes are so vital that without them, cells would not survive for long. But DNA is only part of the story. Proteins, the workers of the cell, also face wear and tear. They fold into precise shapes to function, yet heat, chemical stress, or simple chance can cause them to misfold. Misfolded proteins may clump together, disrupting cellular processes. To prevent this, cells employ chaperone proteins. These molecular guardians help other proteins fold correctly or refold if something goes wrong. If a protein cannot be salvaged, it is tagged with a small molecule called ubiquitin and sent to the proteasome, a structure that functions like a recycling center. There, the faulty protein is broken down into its building blocks, which can be reused to make new, functional proteins. Even the cell's outer boundary, the membrane, is vulnerable. It is constantly exposed to free radicals, mechanical stress, and the wear of everyday exchanges. When lipids in the membrane are damaged, specialized enzymes repair or replace them. Vesicles can deliver fresh components, patching weak spots like tiny repair crews mending a wall. In this way, the membrane remains fluid and resilient, despite facing continuous strain. Repair extends to the cell's organelles as well. Mitochondria, the energy producers, generate reactive molecules as part of their work. Over time, this can damage their own membranes and DNA. Cells monitor mitochondrial health, and when a mitochondrion is beyond repair, they remove it through a process called mitophagy. This targeted recycling prevents damaged powerhouses from causing harm, while ensuring energy production continues smoothly. All of these processes demonstrate a key truth: damage is inevitable, but repair is relentless. Every moment, countless tiny injuries are being corrected, often before they cause noticeable harm. Without this ongoing vigilance, life would quickly unravel under the weight of molecular errors. Interestingly, the efficiency of these repair systems is closely tied to aging. As organisms grow older, DNA repair, protein maintenance, and membrane renewal become less effective. Small errors accumulate, leading to gradual decline. Some researchers believe that understanding and improving cellular repair may hold the key to extending healthy lifespan. It is as though the repair crews slow down with age, leaving the cracks to widen. Repair is not only about fixing, but also about making decisions. Sometimes the damage is too severe to mend safely. In such cases, cells can activate programmed cell death, or apoptosis. This is not a failure, but a form of protection, preventing damaged cells from becoming dangerous, such as by turning cancerous. Apoptosis ensures that the whole organism is safeguarded, even at the cost of individual cells. The quiet repair crews within each cell illustrate resilience on the smallest scale. They show that life is not about avoiding damage altogether, but about managing it with care and precision. From proofreading DNA to recycling proteins and renewing membranes, these systems keep the machinery of life in motion despite constant challenges. It is tempting to think of cells as delicate, but the reality is that they are robust precisely because of these safeguards. Every scar is mended, every broken part replaced, every mistake caught before it becomes too costly. The result is continuity, life carrying forward generation after generation. If balance is the lullaby that sustains cells, then repair is the gentle hand that smooths the sheets and restores order after the inevitable mess. Damage happens, but life is ready. The quiet repair crews never rest, and in their tireless work lies the secret of survival. Every household, no matter how organized, needs regular cleaning. Dust gathers, dishes pile up, and things eventually need to be sorted into what can be reused and what must be discarded. Cells, too, accumulate clutter. Broken proteins, worn-out organelles, and invading debris can all build up if left unchecked. Without a system for cleaning and recycling, the orderly rhythm of life inside a cell would soon dissolve into chaos. Fortunately, cells are excellent housekeepers, armed with mechanisms that not only tidy up but also cleverly repurpose materials. At the heart of this housekeeping lies autophagy. The word comes from Greek roots meaning self-eating, but it is not as grim as it sounds. Autophagy is a process by which cells identify their damaged or surplus parts, wrap them neatly in a double membrane, and deliver them to a specialized recycling center. It is less a feast and more a spring cleaning, where clutter is carefully packed away to be dismantled and turned into useful components. The process begins with the recognition of something unwanted, perhaps a mitochondrion that has grown leaky, or a cluster of misfolded proteins sticking together in a troublesome way. The cell responds by forming a small membrane sac around the item. This sac expands, wrapping fully around its cargo until it resembles a sealed bubble. This bubble, called an autophagosome, is then sent on a journey to meet with another structure, the lysosome. Lysosomes are remarkable organelles, sometimes nicknamed the cell's stomachs or garbage disposals. They are small, acidic compartments packed with enzymes capable of breaking down virtually anything organic. Proteins, fats, carbohydrates, and even entire organelles can be digested inside a lysosome. But this digestion is not wasteful. Instead of discarding the pieces, lysosomes release the raw materials back into the cytoplasm. The cell can then reuse amino acids, fatty acids, and sugars to build new structures or to fuel metabolism. It is a recycling program so efficient that nothing goes to waste. The partnership between autophagosomes and lysosomes forms one of the most elegant cleaning systems in biology. Once the autophagosome fuses with a lysosome, its contents are degraded and recycled. This is how a cell prevents clutter from piling up while simultaneously ensuring a steady supply of resources. In times of abundance, autophagy runs quietly in the background, performing routine maintenance. But in times of scarcity, such as when nutrients are low, autophagy can ramp up dramatically. Cells can survive lean conditions by digesting their own internal reserves, keeping themselves alive until external supplies return. It is important to see this not as destruction but as balance. By breaking down old or damaged parts, the cell makes room for renewal. Much like pruning a tree encourages healthier growth, autophagy ensures that the cell remains efficient and resilient. The old makes way for the new, and the cycle of life continues smoothly. The lysosome itself deserves more attention. It is often overlooked compared to glamorous organelles like the nucleus or mitochondria, but it is absolutely essential. Inside, the environment is acidic, similar to the strength of vinegar. This acidity activates the enzymes, allowing them to digest material effectively. Remarkably, these enzymes remain harmless if they escape into the neutral cytoplasm, preventing accidental damage. It is a safety system built into the design, powerful enough to clean up waste but controlled enough to avoid harming the rest of the cell. Lysosomes are not passive storage lockers, they respond dynamically to the needs of the cell. When large amounts of waste need processing, lysosomes can grow in size or increase in number. When the demand decreases, they shrink or recycle themselves. This flexibility allows cells to handle varying conditions, from normal day-to-day activity to stressful times of repair or starvation. Cleaning is not just about disposal, it is also about defense. When pathogens such as bacteria invade, they may find themselves engulfed and delivered to lysosomes, where they are promptly dismantled. This is an ancient immune mechanism, keeping single cells safe long before complex immune systems evolved in multicellular organisms. In this sense, lysosomes are both janitors and guardians. Autophagy and lysosomal recycling also play roles in development. During certain stages, cells deliberately break down parts of themselves to transform into new forms. For example, as a tadpole becomes a frog, its tail is removed through autophagy. Rather than wastefully discarding it, the material is recycled to support the growth of new tissues. Even at the level of whole organisms, the principle of reuse is central. When these systems fail, the consequences are serious. If lysosomes malfunction, waste products accumulate, leading to diseases known as lysosomal storage disorders. Cells swell with undigested material, and normal function is impaired. Similarly, when autophagy falters, damaged organelles and proteins linger, contributing to neurodegenerative conditions such as Parkinson's or Alzheimer's disease. These disorders highlight how crucial cellular cleaning truly is. Without it, the cell becomes a cluttered space, unable to maintain its health. Interestingly, research suggests that enhancing autophagy might slow aspects of aging. Since damaged proteins and organelles accumulate with time, boosting the cell's recycling pathways could, in theory, help maintain function for longer. Experiments in animals have shown that interventions which stimulate autophagy can increase lifespan. This has led scientists to explore potential ways to harness the process for human health. In essence, A well-kept cellular house may be one of the keys to longevity. On a smaller timescale, the recycling function of autophagy helps cells adapt to changing environments. When energy is scarce, the breakdown of internal components provides a stopgap supply. When toxins accumulate, autophagy helps remove them. It is not a luxury system, but a core survival strategy, always ready to adjust to shifting conditions. The poetry of this process lies in its quietness. Autophagy does not announce itself with fanfare. Lysosomes do not advertise their activity, yet together they maintain order in what would otherwise be a messy, malfunctioning world. They keep the cell clean, efficient and adaptable, transforming waste into resource and clutter into opportunity. Life at the cellular level is not about pristine perfection, but about constant renewal. Nothing is left to rot. Everything is given another chance, whether as raw material for a new protein or fuel for energy production. This gentle cycle of cleaning and recycling is one of the most elegant demonstrations of nature's efficiency. Cells, in their endless work, show a truth that extends beyond biology. Renewal requires letting go of what no longer serves, and even in endings, there are beginnings hidden. The quiet hands of autophagy and lysosomes ensure that life inside the cell never stagnates but flows forward, tidy and resilient. One of the most remarkable qualities of living cells is their ability to adapt to changing conditions. Just as a household might switch from cooking with gas to using electricity when one supply falters, or a traveller might alternate between walking and cycling depending on the terrain, cells too can shift their strategies for generating energy. This is known as fuel flexibility, the capacity to choose between different energy sources depending on availability and demand. For many cells, glucose is the favourite fuel. It is quick to use, relatively easy to transport, and can be broken down rapidly to provide bursts of energy. Imagine glucose as a convenient snack, fast to grab, fast to digest, and always useful when energy is needed urgently. The pathways that metabolize glucose, including glycolysis and cellular respiration, are finely tuned and efficient under normal conditions. But life does not always provide steady supplies of sugar, and cells cannot afford to depend on a single source of fuel. When glucose becomes scarce, cells are ready with a backup plan. Fatty acids, stored in molecules called triglycerides, provide a dense and reliable form of energy. Unlike glucose, which is like a quick snack, fatty acids are more like a slow-cooked meal, rich, sustaining, and capable of fueling the cell for longer stretches. They yield more energy per molecule, though they take more steps to process. Cells equipped with mitochondria are especially adept at burning fatty acids, feeding them into pathways that steadily release energy in the form of ATP, the currency of cellular power. This switch is not random but carefully controlled. Hormones such as insulin and glucagon act like signals at a crossroads, telling cells whether to lean on sugar or fat. When food is plentiful and blood glucose is high, insulin encourages cells to take in and use sugar. When fasting or exercising, glucagon nudges cells toward fatty acid metabolism, drawing on energy reserves stored in fat tissue. This elegant balancing act ensures that energy supply matches both availability and demand, preventing waste while maintaining performance. Fuel flexibility also plays out during physical exertion. At the beginning of exercise, muscles rely heavily on glucose because it can be mobilized quickly. But as activity continues, especially during endurance exercise, muscles begin to rely more on fatty acids. This shift allows glucose to be conserved for situations where speed is critical, while fat provides a more sustainable supply. It is rather like starting a journey with pocket money and then dipping into long-term savings for the road ahead. There are also moments when cells use both fuels at once. This dual strategy is particularly useful in situations of fluctuating energy demand. By keeping multiple pathways active, cells can smoothly transition between energy sources without interruption. It is the biological equivalent of a hybrid car that runs on both electricity and fuel, switching seamlessly between them for efficiency. The liver acts as a key manager in this system, coordinating fuel use across the body. When glucose levels dip, the liver breaks down stored glycogen into glucose molecules that are released into the bloodstream. If fasting continues and glycogen reserves are depleted, the liver begins to produce ketone bodies from fatty acids. These ketones can serve as an alternative fuel for many tissues, including the brain, which normally prefers glucose. It is a clever adaptation that ensures even in prolonged shortages, vital organs remain supplied with energy. This versatility has been shaped by evolution. Our ancestors often faced unpredictable food supplies, with feasts followed by famine. A system that relied solely on glucose would not have been sustainable. The ability to switch between glucose and fat allowed survival during lean times and supported the energy-intensive demands of hunting, foraging, and migration. Modern humans, with more consistent access to food, still carry this flexible machinery within every cell. At the cellular level, the mitochondria play a starring role in this balancing act. These organelles can take in fragments of glucose or fatty acids and feed them into a shared pathway known as the citric acid cycle. This common processing plant allows different fuels to converge into the same energy-making machinery. From the mitochondria's perspective, it does not matter whether the input comes from sugar or fat. What matters is that the end product, ATP, is delivered reliably. Fuel flexibility also provides resilience during stress. In conditions such as low oxygen, cells can temporarily rely more heavily on glucose because it can be broken down without oxygen through glycolysis. This is less efficient, yielding fewer ATP molecules, but it can keep a cell alive during shortfalls. Once oxygen is restored, the cell returns to more efficient pathways, incorporating fatty acid metabolism once again. This adaptability is a survival tool, allowing life to endure under changing circumstances. The trade-offs between glucose and fat metabolism illustrate a broader truth about biology. Efficiency and speed often compete. Glucose is quick, but less energy-dense, while fat is slower but richer. By maintaining the ability to use both, cells secure the best of both worlds, adjusting strategy as the environment demands. Fuel flexibility even extends into more unusual territory. Some cells, particularly in certain microbes, can metabolize other substrates entirely, such as amino acids or even unusual sugars. While human cells rely mostly on glucose and fatty acids, the underlying principle remains the same. Survival depends on the ability to adapt fuel sources to changing conditions. In daily life, this flexibility can be seen in how the human body responds to eating patterns. After a meal rich in carbohydrates, cells are flooded with glucose and eagerly use it. Hours later, as glucose levels drop, fat metabolism takes over quietly in the background. During fasting or prolonged exercise, ketone bodies join the mix. This seamless switching ensures that regardless of circumstances, energy continues to flow without interruption. The elegance of this system lies in its quiet adjustments. There is no dramatic switch, no sudden blackout as one fuel ends and another begins. Instead, the sealed machinery smoothly shifts gears, maintaining continuity. It is a kind of biological jazz, improvising around the theme of energy, always keeping the rhythm steady even as the tune changes. In this flexibility lies resilience. By being able to draw energy from multiple sources, cells are not held hostage by the fluctuations of their environment. They can thrive when sugar is plentiful, endure when fat is the only option, and even invent new strategies when pushed to extremes. It is a lesson in adaptability, life's ability to keep moving forward, even when the pantry shelves look bare. Fuel flexibility shows that survival is not about clinging to a single plan, but about maintaining options. Just as a traveller packs more than one route on a map, the cell carries within it the pathways to switch, blend, and adapt its energy use. In this quiet versatility, life finds the steady power it needs to endure. Life inside a cell is usually calm and steady, with countless reactions flowing in harmony, like a well-practiced orchestra. But every so often, something throws the rhythm off. Perhaps the temperature rises, oxygen becomes scarce, or reactive molecules begin darting around like sparks from a fire. These challenges, known as cellular stresses, are unavoidable. Instead of panicking, however, cells have evolved remarkably gentle and effective coping strategies. They do not collapse under pressure. They bend, adapt, and often emerge stronger. One of the most famous examples of this resilience is the production of heat shock proteins. Despite the name, these helpers are not limited to high temperatures. They come into play whenever proteins inside the cell risk becoming misshapen or tangled. Proteins are delicate structures, folding into precise three-dimensional shapes that allow them to function correctly. Stress, whether from heat, toxins, or other disturbances, can cause these folds to unravel or misalign. Think of it as a library where the books suddenly tumble out of order. The information is still there, but it cannot be accessed properly. Heat shock proteins step in as caretakers, guiding proteins back into their correct shapes or preventing them from clumping together in the first place. They act like patient librarians, reshelving the books so order is restored. Some even escort hopelessly damaged proteins to the recycling centers of the cell, ensuring that clutter does not accumulate. This quiet protection allows cells to withstand bursts of stress that might otherwise prove overwhelming. Another key defense is the use of antioxidants, the molecular equivalents of fire extinguishers. Cells constantly generate reactive oxygen species, tiny, unstable molecules that are natural byproducts of energy production. Under normal conditions, these are kept under control, and in fact, they even serve useful signaling roles. But when their levels rise too high, such as during environmental stress or intense metabolic activity, they can damage DNA, proteins, and membranes. To keep this in check, cells employ a network of antioxidants. Some are small molecules, like vitamin C and vitamin E, that mop up excess reactive oxygen directly. Others are enzymes such as superoxide dismutase and catalase, which transform reactive oxygen into safer forms. Rather than attempting to eliminate every spark, antioxidants maintain balance, ensuring that the occasional reactive molecule does not spiral into a destructive blaze. Stress can also arrive in the form of mechanical strain. Cells are not isolated from the forces of the world around them. They may be stretched, compressed, or pulled as tissues move and shift. To cope, cells reinforce their internal framework, using the cytoskeleton to absorb and distribute pressure. It is similar to how a building might sway gently during a storm yet remain intact, because its design allows for flexibility. Instead of resisting all movement, cells embrace resilience by bending without breaking. Communication plays a large role in coping with stress as well. When a cell encounters difficulty, it often sends out signals to its neighbours. These messages can trigger protective responses in nearby cells, ensuring that an entire tissue or organ is prepared to endure. It is a communal defense, as if one lookout in a village spots approaching danger and sounds the alarm, allowing everyone to take precautions together. The elegance of cellular stress responses lies in their balance. Cells do not overreact with unnecessary force, nor do they ignore the problem. They act with precision, deploying just enough protection to restore stability. Too much reaction could waste energy or cause collateral damage, while too little would leave the cell vulnerable. This measured approach is why stress can be endured without chaos. Interestingly, a small amount of stress can actually be beneficial. Exposing cells to mild challenges often strengthens them, a phenomenon known as hormesis. For instance, low levels of reactive oxygen species can stimulate antioxidant defenses, making the cell better prepared for larger challenges later. This is not unlike training a muscle, where repeated gentle strain builds strength and resilience over time. Heat shock proteins and antioxidants are not flashy solutions. They work quietly in the background, maintaining order while life goes on. They are part of a larger philosophy that cells follow. Do not seek to eliminate all stress, but rather develop systems to cope with it calmly. Because stress is inevitable, survival depends not on avoiding it, but on adapting gracefully. This calm adaptability is why living systems can persist through conditions that seem inhospitable. A rise in temperature, a burst of reactive oxygen, or a tug of mechanical force does not spell disaster. Instead, it becomes another opportunity for cells to demonstrate their resourcefulness. By folding proteins back into place, neutralizing harmful molecules, and reinforcing their frameworks, cells continue their work almost as if nothing happened. What emerges from this is a sense of quiet resilience. Stress is not an interruption of life, but part of the rhythm of existence. Cells respond not with panic, but with steady, practiced measures that preserve balance. It is an elegant reminder that survival often depends not on brute strength, but on the ability to stay composed under pressure. The cell's strategies for coping with stress echo a broader truth. Endurance is built not through resistance alone, but through flexibility, repair, and preparation. Within this calm resilience lies one of life's greatest strengths, the capacity to bend, adapt, and continue thriving, no matter how unpredictable the world becomes. Inside every living cell, time is not just something that passes, it is something that pulses, like a rhythm gently guiding countless processes. Life is not random or chaotic. Instead, it follows cycles, patterns, and internal clocks that ensure order. These rhythms do not demand attention with loud ticks or chimes. They whisper in chemical signals and subtle oscillations. The steady pace allows life to remain in harmony with both its surroundings and its own inner workings. One of the most enchanting rhythms is the circadian cycle. Almost all living things, from simple bacteria to towering redwood trees, keep track of day and night. Cells cannot see the sunrise or watch the stars, but they sense light indirectly through molecules that respond to environmental cues. These molecules tell the cell what time of day it is, guiding when to conserve energy, when to repair, and when to prepare for activity. It is a biological version of a gentle bedside clock, quietly reminding the cell when to rest and when to awaken. For human cells, circadian rhythms are tied closely to sleep, hunger, and energy levels. At night, certain genes become more active, switching on repair systems to tidy up damage accumulated during the day. During daylight, other sets of genes wake up, encouraging the production of proteins needed for energy and activity. These patterns ensure that cells do not exhaust themselves by running every process at once. Instead, they spread out their responsibilities over a 24-hour cycle, much like a city that schedules traffic lights to keep cars flowing smoothly. But circadian cues are only one part of the story. Deep within, each cell also follows its own cycle, the cell cycle. This rhythm determines when a cell grows, when it prepares to divide, and when it carefully duplicates its DNA. The process is measured and deliberate, divided into stages with checkpoints along the way. At each checkpoint, the cell pauses to make sure everything is in order before moving forward. If DNA is damaged or conditions are not right, the cycle slows, allowing time for repair. This patience ensures accuracy and prevents mistakes that could ripple into larger problems. The stages of the cell cycle, growth, synthesis, preparation, and division, form a repeating loop, but not all cells move through it at the same pace. Some cells divide rapidly, like those in the skin or intestines, replacing worn-out neighbours with fresh ones. Others linger in a restful state, content to carry on their duties without dividing for long stretches of time. Neurons, for example, often remain in this restful state for decades, holding on to memories and connections without the need for constant renewal. The balance between activity and rest, division and pause, is itself a rhythm. It is not unlike a piece of music, where notes alternate with silence to create harmony. If the music were all noise without pause, it would become overwhelming. If it were only silence, nothing would happen. In cells, alternating phases of action and stillness give structure to life. Interestingly, rhythms within cells are not isolated. They link with one another and with rhythms of the larger body. The sleep-wake cycle, body temperature, and even hormone release are tied together, creating a multi-layered symphony of timing. When everything aligns, energy flows easily and the body feels in balance. But when rhythms are disrupted, through irregular sleep, stress, or sudden shifts in light exposure, the harmony falters. Jet lag is a familiar example of this misalignment. Cells in different tissues temporarily fall out of step, leaving the body feeling sluggish until the orchestra tunes itself again. Cells also make use of shorter rhythms. Some cycles last only minutes or hours, such as bursts of calcium signals that regulate communication, or pulses of gene activity that come and go like gentle waves. These smaller rhythms may seem less dramatic than the 24-hour circadian pattern or the grand progression of the cell cycle, but they are equally important. They add texture to the overall timing of cellular life, ensuring that processes do not clash or compete for attention. One fascinating feature of biological clocks is their adaptability. While they run on internal timing mechanisms, they can reset themselves when conditions change. Light, food, and temperature all serve as cues that fine-tune the circadian rhythm. A cell does not stubbornly cling to its own sense of time if it conflicts with reality. Instead, it adjusts, ensuring that the individual rhythm fits within the broader pattern of the organism and the environment. The existence of these clocks shows that time is not just something cells passively endure. Time is actively managed, divided, and shaped. It keeps the intricate machinery of life from descending into chaos. Without rhythms, growth might happen at the wrong moment, repair might arrive too late, or energy might be wasted when it is not needed. With rhythms, cells pace themselves, ensuring that every process has its moment. There is a certain calm in recognizing that life is lived in cycles. The rising and setting of the sun, the beating of a heart, the phases of cell growth, all follow patterns that repeat with reassuring familiarity. Within that repetition there is stability. The cell's clocks are not strict taskmasters but gentle guides, reminding life to move steadily forward without rushing. Perhaps that is why rhythms feel so natural and comforting. Just as the body relaxes into the rhythm of breathing, or the cadence of footsteps, cells find balance in their own repeating cycles. The tick of their molecular clocks is not meant to be heard, but to be lived, woven quietly into the fabric of existence. Time inside the cell is not measured by minutes or hours, but by cycles of activity and rest, growth and repair, light and darkness. It is a rhythm as old as life itself, soft yet steady, ensuring that the music of existence plays on without missing a beat. Inside the quiet bustle of a living cell, progress does not happen blindly. Every major step forward is weighed against a set of criteria, as though the cell were pausing at a crossroads, considering whether the road ahead is safe. These are the decision points, checkpoints built into the very fabric of life. They act as guardians of order, ensuring that growth, replication, and division are carried out only when conditions are right. Without them, chaos could unfold, damaged DNA might slip through, resources could be stretched too thin, or a new cell might be born incomplete. The cell cycle is a grand loop of life, moving through phases of growth, DNA replication, preparation, and finally division. But within this loop are several gates. The cell cannot simply stroll past them without proving it is ready. At each gate, molecular inspectors quietly check for mistakes, confirm supplies, and verify that timing is appropriate. If problems are found, the cell does not stubbornly press on. Instead, it pauses, corrects, and only resumes when conditions are satisfactory. One of the most critical decision points is at the very beginning of the cycle, often called the restriction point or G1 checkpoint. Here the cell decides whether to commit to division at all. This is not a trivial choice, because embarking on the journey toward division is resource intensive. It requires duplication of the entire genome, construction of new organelles, and the careful assembly of machinery to divide evenly. If nutrients are scarce, if signals from neighbouring cells discourage growth, or if the DNA shows signs of damage, the cell may instead choose quiescence. Quiescence is not failure, nor is it laziness. It is a restful state, a kind of biological pause button where the cell maintains itself, but does not actively move toward division. Many cells, especially in complex organisms, spend much of their lives in this state. Neurons, for instance, can remain in quiescence for decades, faithfully carrying out their tasks without ever re-entering the cycle. It is a reminder that growth is not always the goal. Sometimes stability is the wiser choice. If conditions at the restriction point are favourable, the cell commits to moving forward. DNA replication begins, and with it comes another crucial checkpoint. During the synthesis phase, the entire genome is copied. Given that even a simple genome contains millions or billions of base pairs, accuracy is paramount. Specialized enzymes not only duplicate the strands but also check their own work, correcting errors as they go. At the end of replication, the checkpoint machinery inspects the finished copies. If errors remain, or if replication is incomplete, the cycle halts until the issues are resolved. The logic here is simple. One does not want to build a new library on shaky foundations. Later, in the preparation phase leading up to division, another gate awaits. This is the G2 checkpoint, where the cell reviews the state of its DNA and ensures it has all the components necessary for division. Imagine a pilot running through a pre-flight checklist: fuel, instruments, weather, engines, before taking off. The cell is no less thorough. If damage is detected or resources are insufficient, the process pauses again, buying time for repair or for external conditions to improve. The final and perhaps most dramatic checkpoint lies at the very heart of division itself. During mitosis, chromosomes must line up neatly before being pulled apart into two daughter cells. This alignment is not optional. If chromosomes are unevenly distributed, one daughter might end up missing critical genes while the other receives extras. To prevent such mistakes, the spindle assembly checkpoint ensures that every chromosome is properly attached to the spindle fibers. Only when the geometry is perfect does the machinery give the green light to proceed. These decision points reveal that the cell cycle is not a reckless dash, but a measured walk through guarded gates. At each step, questions are quietly asked. Is the DNA intact? Are resources available? Are external signals supportive? If the answers are not satisfactory, the cell waits. In this way, the cycle is less like a conveyor belt and more like a series of well-timed decisions. It is worth noting that the choice between quiescence and division is not made in isolation. Signals from neighbouring cells, growth factors circulating in the environment, and the overall state of the organism all influence the outcome. A cell in a healing wound may receive strong encouragement to divide, helping rebuild tissue. A cell in a fully grown organ, by contrast, may be gently told to rest, preserving the structure without unnecessary expansion. These external whispers guide the cell's internal decisions, knitting individual behaviours into the broader needs of the body. The elegance of this system lies in its flexibility. A cell can speed through checkpoints when conditions are excellent, or it can linger when repairs are needed. It can enter quiescence for years, then reawaken if circumstances change. This adaptability ensures survival in a world that is rarely predictable. The checkpoints also serve as guardians against disease. When they function properly, they prevent damaged or unstable cells from dividing. When they fail, errors can slip past and uncontrolled division may follow. This breakdown underlies many cancers, where mutations disable the checkpoints and free the cell from normal decision-making. In a sense, cancer is not a story of too much growth alone, but of lost decision points, cells that no longer pause to ask if they should move forward. Yet in healthy life, the presence of these gates instills calm order. Growth is permitted only when wise, division proceeds only when ready, and rest is always an option. The cell, tiny though it may be, embodies the principle that progress without reflection is dangerous. By building checkpoints into its cycle, it ensures that each step forward is both deliberate and safe. In this way, the decision points of the cell are a lesson in patience. Rather than rushing, the cell embraces the pause, values the checkpoint, and respects the criteria. Each choice balances the needs of the present with the promise of the future, and in those quiet pauses, life maintains its steady, dependable rhythm, one careful decision at a time. In the usual rhythm of cellular life, the story is one of maintenance, growth, and balance. Most of the time, a cell quietly carries out its duties, keeping the organism healthy and stable. But every so often, rare and extraordinary events occur. These moments may seem like exceptions, yet they reveal the deeper rules that guide life. They are the quiet edge cases that show how biology is built, not just a plan for thriving, but also a plan for graceful departure. Among the most striking of these unusual events is apoptosis, often described as programmed cell death. At first glance, the concept may sound unsettling, death woven into the very design of life. But apoptosis is not destruction in the chaotic sense. Instead, it is a carefully choreographed exit, a way for a cell to step aside without causing harm to its neighbours. It is, in many respects, a demonstration of how order prevails, even when endings are involved. Apoptosis unfolds with remarkable elegance. The process begins when a cell receives a signal, sometimes from within itself, sometimes from its environment, that it is no longer needed or that it poses a risk. This might happen if the cell has accumulated too much damage, if its DNA is beyond repair, or simply if the organism needs to reshape tissue. Rather than clinging to life and jeopardizing the whole, the cell quietly agrees to bow out. Once the decision is made, an internal cascade is set into motion. Specialized proteins, often referred to as caspases, act like choreographers, ensuring that the process is smooth and orderly. The cell's contents are carefully dismantled, its DNA fragmented, and its outer shape begins to shrink. Yet even in this disassembly, care is taken. The cell packages its pieces into tidy little vesicles, preventing any leakage of harmful material into its surroundings. Neighbouring cells or immune cells then step in, gently clearing away the remains, almost as though sweeping up after a performance. This controlled departure serves vital purposes. During development, apoptosis helps sculpt the body. The spaces between fingers and toes, for example, are formed because certain cells receive the signal to leave, carving out distinct shapes. In the nervous system, countless neurons are initially created, but many are pruned through apoptosis, leaving behind a streamlined network that functions with precision. Without this built-in editing, the body would carry extra tissue, redundant connections, and structures that could hinder function. Apoptosis also serves as a protective measure. Cells with severely damaged DNA, if allowed to continue, could mutate and cause harm. By stepping aside, these cells protect the larger community. It is an act of self-sacrifice, ensuring the safety of the organism as a whole. In this way, apoptosis highlights an underlying principle, that life is not just about the survival of individual parts, but about the harmony of the entire system. It is worth contrasting apoptosis with another form of cell death, necrosis. Necrosis is accidental, messy, and often triggered by injury or infection. When cells die through necrosis, they burst open, spilling their contents and often triggering inflammation. Apoptosis, by contrast, is neat, intentional, and quiet. It does not draw attention or cause collateral damage. The difference underscores how much energy and planning biology invest in keeping even endings under control. Rare moments like apoptosis also remind us that biology anticipates the unexpected. A cell cannot assume it will last forever. Mutations, stress, or developmental needs may demand a change. By having apoptosis available, life equips itself with a graceful fallback. It is less a sign of weakness than of foresight, an acknowledgement that endings are sometimes as important as beginnings. Other edge cases reveal similar lessons. Consider senescence, a state in which cells stop dividing but do not die. Senescent cells act as a brake. Preventing damaged cells from multiplying further, they remain alive, contributing signals to their neighbours, but they no longer press forward in the cycle. This too is a kind of rare moment, a middle ground between activity and departure. These unusual events, apoptosis, senescence, and others, teach that biology values order and responsibility above unchecked persistence. Cells are not immortal machines driven blindly forward. They are participants in a collective, making choices that benefit the whole. Even in their quiet exits, they demonstrate a commitment to balance. Seen from a broader view, apoptosis is a reminder of how life builds resilience. By planning for rare moments, the system avoids catastrophe. A single cell's departure, handled gracefully, prevents ripple effects that might destabilize the organism. The process ensures continuity, preserving the smooth functioning of tissues and organs. In this sense, apoptosis is not simply about endings, it is a safeguard for ongoing life. There is something almost poetic in this cellular choreography. In everyday moments, cells are engines of activity, building, communicating, and repairing. But in apoptosis, they become performers of a final act, one that embodies humility and foresight. Their departure is not chaotic, but thoughtful, a quiet bow that allows the stage to remain ready for the next performance. These rare moments may not dominate the cell story, but they illuminate its principles. They show that biology is not only about constant activity, but also about recognizing when to pause, when to stop, and when to let go. It is a lesson written in molecular detail. That order sometimes requires departure, and that endings can be as graceful as beginnings. By examining the edge cases, the unusual decisions, and the rare events, one sees the depth of planning woven into life's design. Apoptosis is not an accident of biology, but an essential part of it. It reflects a truth that runs through every level of life, that survival depends not just on persistence, but on knowing when to make way. And in those rare, carefully managed moments, the quiet beauty of order shines most clearly. Life thrives not only because individual cells are remarkably capable, but also because they cooperate across scales, weaving together into tissues, organs, and whole organisms. A single cell, no matter how efficient, can accomplish only so much. But when millions, even billions of cells synchronize their actions, the result is an organism with abilities far greater than the sum of its parts. The story of biology is therefore not only about what happens inside a cell, but about how those microscopic actions scale upward to build living beings. Take muscle tissue as a first example. At the cellular level, each muscle cell contracts when prompted by a signal, pulling tiny filaments of actin and myosin past one another. On its own, one cell's tug would barely be noticeable. Yet when thousands of muscle fibers contract in unison, the tissue generates enough force to bend an arm, carry a load, or even sprint across a field. This is a clear demonstration of scale. The microscopic sliding of protein filaments produces macroscopic strength, movement, and coordination. The cooperation extends further. Muscle cells do not act alone but rely on neighbouring support. They depend on nerve cells to deliver the signals that trigger contraction, and on red blood cells to deliver the oxygen needed to power their activity. Meanwhile, connective tissue provides the scaffolding that keeps muscles anchored to bones. The end result is an organ system, the musculoskeletal system, that turns cellular chemistry into full-bodied action. The liver provides another striking case of cellular cooperation scaling upward. Within the liver, hepatocytes handle an astonishing range of metabolic tasks. Some detoxify harmful compounds, others regulate blood sugar, and still others produce proteins vital for clotting. Each hepatocyte contributes its share, like workers in a vast factory. Together they maintain the body's internal environment with such precision that fluctuations in diet, toxins, or activity are kept in check. If one hepatocyte falters, the effect is minimal. But if the cooperative network breaks down, the consequences ripple outward into the entire body. The nervous system offers yet another level of scaling. A single neuron carries electrical impulses, transmitting information from one point to another. But in isolation, a lone neuron is just a flicker of current. What makes the nervous system extraordinary is the interconnectedness of billions of neurons, each forming thousands of connections with its peers. Networks emerge from this dense web, capable of processing thoughts, generating memories, and coordinating complex behaviors. The leap from electrical spikes within one neuron to consciousness itself illustrates the breathtaking power of cooperation across scales. Even in the lungs, where gas exchange takes place, the principle is clear. At the smallest scale, a single alveolus allows oxygen to diffuse into the blood and carbon dioxide to diffuse out. One alveolus is tiny, yet millions of them working together provide a vast surface area for breathing. Their collective action ensures that with each inhale and exhale, the body receives the oxygen it needs and expels the waste gases it cannot keep. The smooth rhythm of breathing depends on countless cooperative exchanges at the cellular level. This layered cooperation also explains resilience. In tissues, if one cell is damaged, others around it can often compensate, keeping the organ functional. Redundancy is built into the system, a safeguard against the unpredictability of life. The heart, for instance, contains specialized pacemaker cells that generate rhythmic contractions. If some pacemaker cells fail, others can take over, ensuring that the heartbeat continues. The system does not collapse at the first sign of trouble because cooperation distributes responsibility. Cooperation across scales is not limited to physical actions, it extends to communication. Hormones illustrate this beautifully. A gland releases a chemical signal into the bloodstream, and countless target cells across the body respond in harmony. One tiny gland in the neck, the thyroid, can influence metabolism in nearly every cell. This is cooperation in reverse, a large-scale signal coordinating the behavior of the smallest units. The flow of information between scales ensures that the body acts as one cohesive whole, rather than as a collection of independent parts. Another dimension of cooperation is evident in the immune system. At the cellular level, immune cells detect, tag, and neutralize threats. Some cells specialize in recognizing invaders, while others produce antibodies, and still others coordinate the attack. Alone, one immune cell could do little to protect the body. Together, however, they create a dynamic defense force, adaptable and resilient. The cooperation spans scales. Cells gather local intelligence, tissues amplify responses, and the organism as a whole mounts a defense that keeps pathogens at bay. There is also the remarkable example of wound healing. When the skin is cut, cells at the site begin dividing rapidly to replace the lost tissue. Platelets form clots, fibroblasts build new connective tissue, and immune cells clear debris. Each participant has its role, yet the outcome is collective, the restoration of the skin's barrier. The choreography of so many different cell types demonstrates that cooperation is not optional, it is the very basis of repair and recovery. Zooming out further still, the concept of cooperation across scales applies to ecosystems and communities of organisms. The cells of one organism support tissues and organs, but organisms themselves cooperate to form ecosystems. A forest, for example, depends on trees, fungi, microbes, and animals all interacting. The principle echoes at every level of life. Survival is achieved not through isolation, but through interconnection. What makes this cooperation so effective is the balance of autonomy and integration. Each cell retains a measure of independence, carrying out its core processes, yet each is also responsive to larger signals, adjusting its behavior to fit the needs of the tissue or organ. This balance allows both flexibility and coordination, ensuring that life is adaptable without descending into chaos. The theme of cooperation across scales is a reminder that biology builds order from collaboration. Small actions, like a cell burning glucose, contracting filaments, or releasing a molecule, scale upward into actions as grand as running, thinking, or breathing. Without cooperation, the body would dissolve into disjointed fragments. With it, cells combine to form tissues, tissues form organs, and organs sustain an organism capable of thriving in the world. The quiet beauty of this cooperation is that it often goes unnoticed. Breathing happens without thought, the heart beats without conscious effort, and wounds heal without instructions. Beneath the surface, however, millions of microscopic participants are at work, contributing to a shared purpose. It is through their collective effort that the body achieves harmony. By tracing the path from cell to tissue to organ, it becomes clear that scale is not a barrier but a bridge. The smallest units of life are never insignificant. They are the foundation upon which all else is built. Cooperation is the thread that binds the levels together, ensuring that life functions smoothly from the tiniest reaction to the broadest motion. And in this layered fabric of collaboration, the remarkable truth emerges. Greatness is built not from solitary power, but from the quiet coordination of countless small contributions. A thought, a memory, a shiver down the spine, or the simple act of lifting a finger, all of these begin as tiny signals inside cells. At the smallest scale, the movements are electrical sparks and chemical messengers darting across membranes. At the larger scale, they become sensations, actions, and even the continuity of identity. Bridging the microscopic to the macroscopic reveals how the world inside cells gives rise to the experiences that shape life. Consider something as ordinary as reaching out to grab a cup of tea. The process begins before muscles ever move. Neurons in the brain send signals to one another, relaying intent and preparing a command. At the cellular level, this command is carried by the movement of ions like sodium and potassium, racing through channels in a neuron's membrane. The sudden change in electrical potential creates an action potential, which travels along the length of the neuron like a wave. This is not a single signal, but a carefully timed sequence, a cascade that maintains its strength until it reaches the end of the neuron. At the synapse, the tiny gap between one neuron and the next, the electrical impulse is translated into a chemical one. Vesicles filled with neurotransmitters fuse with the membrane and release their contents into the space between cells. These molecules drift across the gap and bind to receptors on the neighbouring cell, triggering it to fire in turn. Billions of such synaptic exchanges occur in every moment, forming the neural chatter that allows thought to turn into action. When the signal reaches motor neurons that extend into the muscles, the scale of action begins to shift. Motor neurons release a neurotransmitter called acetylcholine, which binds to receptors on muscle cells. In response, the muscle cell opens ion channels, allowing calcium ions to flood inside. These ions set off a series of molecular events. Filaments of actin and myosin slide past one another, contracting the muscle fibre. Multiply that contraction across countless fibres, and the hand rises. What began as the quiet flicker of ions has become the visible action of reaching. The transformation of signals into sensations follows similar principles. Take the experience of hearing music. Sound waves enter the ear and set tiny hair cells in the cochlea vibrating. Each vibration opens ion channels in the hair cells, generating electrical signals. These signals travel through the auditory nerve to the brain, where patterns are interpreted as rhythm, melody, and harmony. At the root, the sensation of music is nothing more than ion movements and neurotransmitter release. Yet at the larger scale, it is an emotional experience capable of stirring joy or nostalgia. Memory consolidation offers another example of how microscopic processes scale upward into complex human functions. When learning occurs, neurons strengthen their connections with one another. At the cellular level, this involves the repeated release of neurotransmitters and the insertion of additional receptors into synapses. The process is called long-term potentiation. Over time, these strengthened synapses become the scaffolding upon which memories are built. The chemical reshaping of synapses eventually stabilizes into structural changes, allowing the memory to persist. Thus, the fleeting act of neurotransmitter release can lead to the enduring ability to recall a childhood home or a friend's voice. The immune system, too, shows how small signals become larger outcomes. When a pathogen enters the body, immune cells release signaling molecules called cytokines. These molecules influence other immune cells, recruiting them to the site of infection and ramping up defensive actions. The sensation of fever or fatigue is partly the result of these signaling cascades. The micro cause, cytokine release, scales upward into the macro feeling of illness, which encourages rest and recovery. Pain is another case where signals become sensations. Specialized nerve endings called nociceptors detect harmful stimuli. When triggered, they send electrical signals through neurons to the spinal cord and brain. Along the way, neurotransmitters like glutamate and substance P amplify the message. At the conscious level, the result is the sharp sensation of pain, a powerful motivator to withdraw from danger. Without the microscopic channels and messengers, the larger experience of pain would not exist, and survival would be jeopardized. Even emotions are rooted in cellular signaling. Feelings of calm or stress are tied to the release of neurotransmitters and hormones. For example, serotonin contributes to mood regulation by influencing how neurons communicate in regions of the brain involved in emotion. Dopamine, another neurotransmitter, shapes motivation and reward. The movements of these molecules across synapses, measured in trillionths of a gram, scale upward into the fullness of human feelings: delight, anticipation, even love. Timing plays a crucial role in how signals become sensations. In muscle contraction, the precise release and removal of calcium ions ensures that contractions are smooth and coordinated. In memory, the timing of signals between neurons determines whether a connection strengthens or fades. Mistimed signaling can lead to disruptions, a muscle twitch, a missed memory, or in more serious cases, neurological disorders. Harmony in sensation and function depends on rhythm in the cellular symphony. The beauty of this system is its adaptability. Neurons and muscles can adjust their sensitivity, becoming more responsive with use, or less so when overstimulated. This plasticity allows for learning, training, and recovery. Practice on a musical instrument, for instance, strengthens neural pathways, turning halting notes into fluid melodies. At the cellular level, receptors and ion channels adjust their numbers and efficiency. What emerges at the macro level is skill and memory encoded in the body itself. Even rest is influenced by microsignals. During sleep, neurotransmitters like GABA quiet neural activity, allowing the brain to enter restorative states. At the same time, during certain phases of sleep, bursts of neural activity help consolidate memories. The larger feeling of waking refreshed or groggy is deeply tied to these cellular processes. The difference between rest and exhaustion can come down to whether molecular signals aligned properly overnight. It is tempting to think of sensations and functions as belonging only to the realm of the organism, but at every step they are built upon cellular exchanges. The gentle flood of calcium in a muscle fibre, the binding of a neurotransmitter to its receptor, the reinforcement of a synapse. All of these are the quiet groundwork for action, thought, and feeling. The smallest sparks in the cellular world ignite the brightest experiences in the conscious one. This mapping from micro to macro emphasizes the unity of biology. Signals do not vanish when they scale upward. They accumulate, resonate, and transform. The cascade is seamless. An ion moves, a channel opens, a neurotransmitter is released, and suddenly an idea is remembered, a song is heard, or a hand is lifted. The wonder lies in the fact that sensation and action, often taken for granted, are rooted in processes so subtle they would pass unnoticed without magnification. By tracing these pathways, the connection between cellular signaling and lived experience becomes clear. At the foundation are molecules and ions, their movements choreographed with precision. At the peak are sensations, movements, and memories that define what it means to live. The journey from signal to sensation is not merely a chain of events, It is the bridge that links biology to experience, and the proof that within each cell's quiet activity lies the story of the body's grand performance. When the lights of the day fade and the body settles into rest, an equally important shift begins within the cells. While it might seem as though everything slows into silence, the nighttime is anything but idle. Cells turn from the rush of daily activity toward a quieter, restorative mode. This is the night shift, a time for repair, recycling, and careful housekeeping. The pace is slower, the rhythm more measured, but the work is no less vital. During waking hours, cells are busy juggling immediate demands, fueling muscles, responding to signals, and producing proteins at high speed. These tasks inevitably generate wear and clutter. Damaged molecules accumulate, proteins fold imperfectly, and tiny reactive fragments called free radicals are left behind. The nighttime offers a chance to sweep up this mess and restore order. Without this daily rhythm of maintenance, cells would gradually drown in their own byproducts. One of the most important nighttime processes is DNA repair. Every day, DNA suffers from small insults, radiation from sunlight, mistakes during replication, and chemical byproducts of metabolism. Most are minor, but over time they could add up to mutations if left unattended. At night, when cells are less preoccupied with division or responding to external stress, specialized enzymes patrol the DNA, scanning for nicks, mismatches, or broken strands. Repair proteins cut out damaged bases, patch them with the correct nucleotides, and seal the chain back together. This quiet routine keeps the genetic script intact so that the instructions for tomorrow's activities remain reliable. Another major player in the night shift is autophagy, which literally means self-eating. Despite the slightly ominous name, it is a remarkably gentle process. Cells enclose worn-out proteins and organelles in double-membraned vesicles, which then fuse with lysosomes, the cell's recycling centers. Enzymes inside the lysosomes break the components down into raw materials. Amino acids, lipids, and sugars are salvaged, ready to be reused in the morning. Instead of discarding waste, the cell transforms clutter into resources, a highly efficient system that resembles a recycling plant running at full efficiency while the city outside sleeps. The metabolic tempo also changes as the night shift begins. Energy demand is lower because the body is at rest, so cells reduce their burning of glucose and fatty acids. Instead, energy production is tuned to be steady rather than intense. Mitochondria, the power stations of the cell, generate ATP at a calmer rate. This slower cadence reduces the buildup of free radicals, which means less oxidative stress. It is as if the engines of the cell are idling smoothly rather than revving at high speed, conserving both resources and structural integrity. Hormones guide this transition into nighttime maintenance. Melatonin, secreted as darkness falls, signals cells to adjust their rhythms. Growth hormone, released in pulses during sleep, supports tissue repair and protein synthesis. These signals act like the orders from a night manager, shifting the priorities of the workforce toward healing and rebuilding. The balance of cortisol, the daytime stress hormone, also drops overnight, further encouraging a restorative environment. In muscle cells, nighttime brings recovery from the strain of the day. Microtears created during activity are repaired as proteins are synthesized and organized into stronger fibers. In the liver, detoxification processes peak, breaking down toxins and metabolizing substances accumulated during the day. In the brain, neurons undergo a kind of cleaning as well. Cellular waste is flushed more efficiently during sleep thanks to the glymphatic system, which clears away byproducts like beta amyloid that could otherwise build up. This is why deep sleep feels so restorative, because the cleanup crew has been given time and space to do its work. The night shift is also a period of recalibration. Ion channels reset their gradients, membranes repair subtle damage, and signaling receptors are recycled or replaced. These resets ensure that cells are sensitive and responsive when the next wave of daylight activity arrives. If the night's maintenance is disrupted by lack of sleep, poor nutrition or stress, the consequences ripple outward. Energy falters, concentration weakens, and long-term health can be compromised. The value of rest lies not just in quieting the mind, but in enabling the cellular world to restore itself. Interestingly, not all cells keep the same pace during the night. Some immune cells become more active after dark, patrolling tissues and identifying potential threats. This nocturnal vigilance may explain why symptoms of illness like fever or congestion often worsen at night. The immune system is more engaged, releasing signaling molecules that promote healing but also cause discomfort. This is another reminder that nighttime is not a shutdown but a reallocation of resources. Autophagy, in particular, deserves another glance because of its elegance. By recycling worn components, it also prevents cells from becoming too cluttered. Imagine a room where nothing was ever thrown away. Eventually, even walking would become impossible. Autophagy prevents this scenario, keeping the cytoplasm spacious and functional. Beyond simple tidiness, it also serves as a survival strategy. In times of fasting or stress, cells can break down their own components to generate energy, allowing them to endure lean times. Nighttime often overlaps with fasting, so autophagy naturally increases, linking sleep, metabolism, and cellular longevity. The overall effect of the night shift is a kind of reset. The day may wear cells down, but the night restores them. Mitochondria generate ATP with less stress, proteins are refolded or recycled, DNA is patched, and membranes are repaired. The entire system emerges refreshed and calibrated, ready to meet the demands of a new day. This is why disruption of the sleep cycle, through irregular hours, shift work, or prolonged wakefulness, can feel so profoundly disorienting. Without the night shift running smoothly, the balance between activity and restoration is thrown off kilter. There is a poetry in how cells use darkness not as a pause but as an opportunity. Instead of seeing the night as an absence of action, it is better understood as the hidden half of life's rhythm. What appears as rest is in fact maintenance, an intricate choreography of recycling and repair that sustains the whole organism. From the DNA double helix to the mitochondria, from lysosomes to the sweeping flows of the glymphatic system, the night shift ensures that the microscopic workers are ready for another sunrise. The body, in its quiet wisdom, has divided the labour of living into day and night. Daytime is for interaction, effort, and output. Nighttime is for healing, tidying, and preparing. Together they form a cycle of activity and restoration, each essential to the other. By appreciating the importance of the night shift, the silent work of cellular caretakers comes into focus, reminding us that the rhythms of rest are as crucial to survival as the bustle of the day. Looking closely at a single cell can feel like gazing at a bustling metropolis, but step back far enough, and the view changes into something even more profound. The story of deep time. The features that make a cell tick are not arbitrary. They are the outcome of billions of years of refinement, trial, and survival. Every pump, every enzyme, every quiet feedback loop carries with it the weight of history. To understand why a cell looks and behaves as it does today, it helps to zoom out and see the long view, the evolutionary journey that etched these systems into existence. One of the most striking realizations is just how conserved many pathways are. The same molecular logic guiding energy production in human cells also operates in yeast, in plants, and even in bacteria. Glycolysis, the 10-step pathway that breaks glucose into smaller pieces, is practically universal. Whether inside a muscle fibre powering a sprint or in a microbe thriving near a hydrothermal vent, the same steps are carried out by similar enzymes. The fact that this pathway is so widely shared suggests it is ancient, dating back to some of the earliest forms of life. Once evolution stumbled upon an effective way to release energy from sugar, the blueprint proved too efficient to abandon. The mitochondrion provides another glimpse into history. These energy factories are thought to have once been independent bacteria, swallowed by ancestral cells more than 1 billion years ago. Instead of digestion, a partnership formed. The host cell provided protection and raw materials, while the engulfed bacterium offered a superior way to generate energy. Over countless generations, genes were exchanged, roles became intertwined, and what was once a merger became a single organism. Every time a mitochondrion spins up to produce ATP today, it is reenacting an ancient alliance. The echoes of that cooperation are still carried in the tiny loop of mitochondrial DNA, a relic of its former independence. Other cellular systems trace back even further, to the earliest chemical reactions that predated life. Proton gradients, the separation of positive charges across a boundary, are one example. Modern cells use them to power ATP synthase, the rotary enzyme that generates ATP. Yet the principle of using a gradient to drive work may have existed before life itself, in natural settings like alkaline hydrothermal vents, where minerals formed porous structures. These natural environments could have created primitive proton gradients, offering the first hints of energy harnessing. Today, every cell continues the tradition, building and maintaining its own gradients as if honouring the original spark of bioenergetics. The universality of the genetic code is another testament to shared ancestry. With very few exceptions, the same triplets of nucleotides specify the same amino acids across the entire spectrum of life. This means that a gene from a jellyfish can be placed into a bacterium, and the bacterium will still produce the jellyfish protein. Such fidelity across vast evolutionary distances underscores how early the code was fixed. Once the mapping between nucleotides and amino acids was established, it became the foundation upon which everything else was built. Changing it would have been catastrophic, so the code endured. Even mechanisms of repair and stress response have roots stretching deep into history. Heat shock proteins, for example, are found in organisms from simple bacteria to humans. Their role as molecular chaperones, helping proteins fold correctly under duress, is so vital that evolution preserved them across eons. Similarly, enzymes that correct DNA mistakes exist in even the most primitive microbes. It is as though evolution recognized early that preserving information and protecting structural integrity were essential for continuity, and those strategies were locked in. But evolution is not only about conserving the old, it's also about innovation layered on top of sturdy foundations. Consider photosynthesis. The earliest versions of life could extract energy from simple chemical reactions, but some organisms evolved a remarkable new trick, using sunlight itself as fuel. Cyanobacteria pioneered this ability, and in doing so, they transformed Earth. They released oxygen as a byproduct, reshaping the atmosphere and paving the way for complex aerobic life. The oxygen-breathing cells of today carry within them that revolutionary shift, still dependent on a planet enriched by an ancient microbial invention. The cell cycle itself, the sequence of growth, replication, and division, is another example of both conservation and adaptation. The fundamental idea of checkpoints and orderly progression is shared across eukaryotes, but the details have been refined in different lineages. This balance of old and new is what gives evolution its texture, a combination of deeply rooted continuity and creative modification. Tracing cellular history also reveals the power of cooperation. Symbiosis is not just the story of mitochondria, but a recurring theme. Chloroplasts, the green photosynthetic machines of plant cells, also began as independent bacteria. Even today, microbial communities exchange nutrients and signals in ways that echo larger-scale ecosystems. Evolution has shown repeatedly that collaboration can be as powerful a survival strategy as competition. Cells bear the imprint of those ancient compromises, proving that life is not only about struggle but also about partnership. Looking at cells with this long view also brings humility. The features often taken for granted, ATP production, membrane channels, protein folding, are not trivial conveniences. They are ancient victories, hard won through trial and error in environments that were far harsher than anything most life encounters today. Each system that persists is a survivor, honed by selection and resilient enough to be passed forward unbroken across billions of years. It also highlights the continuity between all forms of life. Humans may seem distant from bacteria, yet the core of cellular machinery is shared. The enzymes that churn in human cells have recognizable cousins in microbes. The rules that govern signaling, repair, and replication are written in a common biological language. This shared heritage is why studying yeast or worms can reveal truths about human health. The unity of cellular processes reflects a single origin, branching outward into the rich diversity of species seen today. The long view also adds perspective on fragility. Evolution built robust systems, but not perfect ones. Errors still occur, aging still accumulates, and vulnerabilities still exist. These imperfections are not flaws of design but traces of history. Evolution works with what is available, tweaking and adapting rather than starting from scratch. The result is a patchwork of solutions, many elegant, some clunky, but all functional enough to persist. Recognizing this helps explain why some cellular mechanisms seem surprisingly convoluted. They are the descendants of ancient improvisations that simply worked well enough to last. From the first sparks of metabolism to the finely tuned symphonies of modern multicellular life, cells carry the memory of evolution's journey. Every conserved pathway is like a fossil written in chemistry, telling a story that predates species, continents, and even the atmosphere as it exists today. By tracing these stories, one sees not just the machinery of life, but its history, written in enzymes and gradients, in codes and cycles. The long view reveals that a cell is not simply alive in the present, it is a living archive, a thread connecting the deep past to the unfolding future. The image of a shoreline has followed throughout this quiet journey, and it feels natural to return to it now. A cell, at its simplest, is bound by a membrane, its own shoreline, where the inside world meets the outside. That thin boundary is not a wall, but a living edge. Molecules drift across it like travellers arriving and departing, guided by currents and channels, much like the ebb and flow of waves along the sand. It is at this edge that life both protects itself and reaches out, both defines its space and participates in something larger. Every cell is a little harbour, holding within it treasures of order while still touching the sea of its surroundings. Proteins stand like quiet gatekeepers at this boundary, deciding who enters and who leaves. Lipids shift and flex, never rigid, always fluid like water itself. Signals knock, nutrients arrive, waste drifts away, all part of the rhythm that keeps the shoreline alive. To picture the cell without this membrane is impossible. It would be like imagining the tide without a coast. Across this living shoreline, balance is maintained. Gradients ripple like subtle tides, with ions moving in and out, creating differences in charge and concentration. The hum of these movements is soft but steady, almost like a background pulse. In fact, the beating of a heart, the gentle rise and fall that sustains life, owes its rhythm to cells conducting their signals across these very boundaries. Sodium and potassium, calcium and chloride. Tiny travellers ferrying messages across a membrane shore, giving rise to sensations, to muscle contractions, and to the measured hush of a heartbeat. The metaphor circles back here with purpose, because the life of a cell is not a straight line, but a cycle, a tide, a rhythm. Each day brings stresses and repairs, signals and silences, stretches of activity and rest. At night, when the body turns towards stillness, the cells do not stop. Instead, their tempo changes. Metabolism slows, repair takes priority, and the night shift of molecular caretakers steps forward. The shoreline of the membrane remains, but the waves crash more softly. Nutrient intake is reduced, damage is patched, and recycling pathways tidy what is no longer useful. The hush is not empty silence. It is the murmur of maintenance, the background work that prepares for tomorrow's sunrise. This quiet activity echoes the larger cycles that the whole body obeys. Just as the moon tugs the ocean tides, circadian rhythms sweep through tissues, reminding cells when to rest and when to rise. Hormones ebb and flow in predictable patterns, aligning metabolism, repair, and immune defenses with day and night. Every cell along its shoreline listens to these cues, syncing itself to the larger body, to the larger world. Even in rest, there is connection. The shoreline is also a place of reflection, both literal and figurative. Waves pull back what is no longer needed, carrying fragments away. Within the cell, lysosomes and autophagosomes play the same role, breaking down damaged proteins and recycling their parts. It is as though the tide cleansed the beach each night, leaving the sand smooth again for morning. The system is not wasteful. Even in cleaning, it conserves, repurposing what can be used anew. Out of what is broken, life builds again. And still, beneath all of this complexity, the heart keeps its hush. Each thump is powered by countless ion channels, opening and closing, by cells exchanging tiny sparks across their membranes. It is easy to forget that what feels like one heartbeat is actually the orchestration of billions of cells keeping time together. Each shoreline contributes its rhythm, and together they become a steady drumbeat, the sound of life itself. The hush is not silence, but harmony. The long view reminds that these rhythms are ancient. The first cells, adrift in primordial seas, also had boundaries, also maintained gradients, also balanced order against entropy. Their membranes were shorelines too, defining the self against the vastness of the world outside. In each heartbeat today, there is an echo of those first pulses of chemistry, a continuity that stretches back billions of years. The cell's shoreline is not just a boundary, it is a connection to deep time. Standing again at this metaphorical shore, one sees the recurring patterns. Boundaries and exchanges, stress and repair, activity and rest, all part of a cycle that sustains and renews. The shoreline is never static, but it never disappears. It is always there, shifting with the tides, shaping the rhythm, allowing the cell to be both itself and part of something larger. And as the arc settles here, it feels right to rest with the imagery of the tide at its gentlest. Waves roll in, then slide back with soft cadence. The boundary between cell and world hums quietly, like the hush of a heart in the stillness of night. The shoreline is calm, the work of the day done, the repairs underway, the rhythms intact. Life continues its quiet maintenance, unseen but unbroken. In the end, the shoreline of the cell is not only about separation but also about continuity. It marks where life touches the world, where inside meets outside, where individual rhythm blends with greater cycles. Like the coast meeting the sea, it is both edge and beginning, and in its steady pulse, in its gentle hush, it offers a reminder. Life persists, balanced at the water's edge, cradled by the quiet rhythm of the tide. The journey slows, like a tide easing into stillness. The patterns continue, even in rest. The cells do not stop their quiet choreography, and neither does the body they sustain. Tonight has been a gentle unfolding of hidden rhythms, a reminder that even the smallest parts of life move with grace and balance. 3 threads remain to be held softly. First, that life is built on harmony between inside and outside, on boundaries that are never rigid but always fluid, like shorelines touched by the sea. Second, that balance is not stillness but a steady exchange, feedback loops humming along like a lullaby, ensuring stability without ever freezing in place. And third, that repair and renewal are not interruptions to living, but part of it, as natural as the turning of day into night. With these ideas in mind, the patterns can be trusted to continue without effort. The cell does not need permission to keep working, nor does the body need constant supervision. Even in sleep, the signals flow, the repairs take place, and the tides of metabolism keep their rhythm. Curiosity can pause here, resting gently in the knowledge that the story of life carries on quietly, dependably, beautifully. The waves of thought can settle now, drifting into the same cadence as the heartbeat hush. The shoreline is calm, the work is ongoing, and there is nothing more to do. Sleep belongs to this moment, and the patterns will be waiting, still flowing when waking comes again. Thank you for being here and for sharing this quiet journey. If you enjoyed it, a like and a subscription help more than you might imagine. They let these explorations keep growing and make sure you don't miss the next one. And if there's something you'd like me to cover, another hidden pattern of life, a mystery of science, or a corner of the world you'd like to wander through, let me know in the comments. This channel grows best when curiosity points the way. Until then, may your rest be gentle, your dreams unhurried, and your wonder never far away.